नको नको मोहिते सरांनी घेतलं होतं ना.
You cannot start your video because the host has stopped it. Okay. Start. Okay. Yes. So I welcome all the participants for the Foundation Day lecture series. As you know, I think everybody knows that uh, today is our 39th Foundation Day. And on account of our Foundation Day, we have organized one webinar on entrepreneurship development for sustainable fisheries. From morning, just to give you the information about the today's happening, since yesterday evening, College of Fisheries is without electricity. There was a little cyclone, and because of that, electricity is not there. Still, it has not come. But still, we have started with the program with the great difficulty. If at all there is any break in this one, kindly understand that this is maybe due to the failure of electricity or failure of our data range. So please bear with us. It is because of our environment which has been changing. As you know, in the Konkan region, this particular period that is July and August, is with the heavy rainfall, and therefore, please bear with us. Also, there is one change in the program, as our Honorable Vice Chancellor and other directors, they are in one meeting. So they will be joining at 3 o'clock. So our inauguration program, we are just postponing for some time, and we are starting with the first lecture. And first lecture is by Mr. Baneshwar Singh. Okay, so I welcome you, all of
Dr.
you,
Chaudhary, I have
for
already joined. Huh?
the
Dr. Chaudhary, I have joined.
39th Foundation Day lecture series with the
Dr. Chaudhary, I have joined.
webinar on, yes.
Dr. Chaudhary, I have joined. Huh?
Yes, sir. So we are starting with the first lecture by the Baneshwar Singh, and he will be elaborating us on the bugs and bacteria to the rescue of forensic and fisheries science. Before he starts, I will just give the introduction. See, the Baneshwar Singh, he has done BFSc from College of Fisheries Ratnagiri, and we are very proud of him that he is the student from College of Fisheries Ratnagiri and now has reached to the such a height. Now he is working as associate professor at Virginia Commonwealth University, Richmond, in the Virginia. His research interests revolve around the application of genomic tools in the forensic entomology, forensic biology, and forensic microbiology pathology. Specifically, he is interested in ecology and evolution of arthropods and microbial communities associated with both living and dead organisms and their forensics applications. Arthropods and microbes he gathered from human and animal remains for the estimation of minimum post-mortem interval. Forensically, understanding these ecological relationship between the arthropods and the microbes may prove a greater insight into the larval development and consequently increase our ability to provide more accurate estimations concerning the timing of initial colonization of human remains. Microbes associated with the living humans can help in development of new methods for the forensic identification of biological samples, human individualizations based on microbial signatures, as well as the trace evidences analysis. Mr. Baneshwar Singh had his professional positions as an associate professor in the Department of Forensic Science, Virginia. Now you may ask, sir, so why in the fisheries the forensic science? It is because Baneshwar Singh has completed his BFSc from College of Fisheries Ratnagiri, MFSc from CIF Mumbai, and then PhD from CIF Mumbai, and then he had his career in the forensic science only after going to the USA, and there he discovered from fisheries science he diverted to the forensic science. So he worked as he is now working as an associate professor. Earlier he has worked as an assistant professor from August 2013 to near about June 2020, and from June 2020 now he is working as an associate professor. He had his postdoctoral research associateship in the Department of Entomology in Texas. His postdoctoral advisor was Dr. Jeffrey Tomberburn and the Aaron Teron. His project title was The Development of Standard Operating Procedures for Measuring Microbial Populations for Estimating the Post-Mortem Interval. Okay, for that he got the funding from the National Institute of Justice. He had his graduate teaching and research assistantships at the Virginia University during 2005 to 2011. And he also worked as a laboratory manager for the genomic core facility in the he also had the junior research fellowship of the ICR UGC at the CIF Mumbai. Earlier I told you that he had his education PhD in biology from Department of Biology, West Virginia University, and his PhD advisor was Dr. Jeffrey D. And his dissertation title was Molecular Phylogeny of Australia. He had his MFSc in Fishery Resource Management. He got the gold medal. In BFSc also he got the gold medal, and he was a university topper. He has published many papers, many research papers. And therefore we have a very towering personality for our first lecture as a foundation 39th Foundation Day lecture for College of Fisheries, Ratnagiri. I welcome Dr. Baneshwar Singh, and I now request him to kindly deliver the first lecture of our 39th Foundation Day.
Light loud.
Okay, thank you, Dr. Chaudhary, and all my teachers from College of Fisheries, Ratnagiri, and my friends from my class. It's really nice to talk to you all today, and it's early 4:30, and we have similar situation. It's storm here, and state is closed today because of a storm warning. But we are lucky we don't have problem with.
Later issue killers.
Yeah, we don't have any issue with electricity or internet, so we are good to go. And as Dr. Chaudhary sir said, like I did my undergrad and master in fisheries, and then I moved to forensic, but did not leave fisheries. I still have interest in fisheries, and many techniques I use is common for all disciplines. And it's basically the move was also because of the subject. It's all because I studied crustaceans in my master, and then I moved to arthropods and then to microbes. So it's all connected and it's now multidisciplinary. So what I will do today is I will talk about what is my research and where this research can be utilized for fisheries. Excuse me, it will be more forensic heavy. And I know that some of you want to listen more about fisheries, but I think the techniques I will talk will be useful for broader audience. So the topic for today, I will talk about bugs and bacteria and how we can use it to help forensic and fisheries science. And before we start, I just want to warn that there are some slides that have some graphic content. So, if you have issue with dead bodies and things like that, then make sure that you don't get disturbed, because there will be some slide where we have those graphic images. So why bugs and bacteria? The first question that we have, why bugs and bacteria? As you can see the graph here, more than 50% of all animal species on Earth are insects or bugs, right? We have other vertebrates, we have mollusks, crustaceans, and other, invertebrates and vertebrates, but more than 50% are bugs. And if you see bacteria, it's only 1% in terms of what we know, but majority of them we don't know. More than 100 million species are there, and more than 99% we don't know. So the problem is we have a lot, but we don't know many of them. So these two are the most, dominant species on Earth. If you see in the microbes on our body, we have almost half of our cells that are of bacterial in origin. So almost 39 trillion bacterial cells and 30 trillion human cells. So we have lots of bacteria, and my research is mainly focused on those bacteria, how we can use them for our beneficial purposes. If you see our human stool, it has almost 50% by weight of bacteria. So we have lots of bacteria, especially in fecal matter, we have lots of bacteria. So what my research is focused on is between insect, microbes, and how they interact. So I put it here, necrobiome includes all community of organisms associated with decomposing remains. As you can see, we have the dead body and then it interacts with lots of epinecrotic organisms, mostly microbes, then there are blowflies that goes to the dead body, and then beetles and then scavengers. All of them use this for the decomposition purpose. We can use these bugs for PMI prediction. So how we can use? Initially, when I started working, I did not know these bugs. I was not sure what. When my PhD mentor said, okay, get some blowflies, I did not know what blowflies are. And then I started searching, and then I came to know it has lots of application, but I did not give attention before starting the study. So when we did the blowfly can be used for PMI prediction. Not only that, in many situations you have no other way but to just use fly to tell how old the dead body is. Like for example, right now also there are several cases going on where it's very critical to know when someone died. Within 36 to 48 hours, medical examiner and doctors can tell based on different algar mortis, ragar mortis, and things like that. But after that, after 2 or 3 days, sometimes you have no other way but to use bugs to tell how old the dead body is. Generally, we use two methods. One is insect development data. That means when the fly comes to dead body, they lay egg, they develop to different stage of larvae, and then they turn to pupa and adult. As you can see, this cycle depends on temperature. So when we have any remains and we get some evidence, we can go backside and we can calculate how old this insect is. And that tells us how old the dead body is. So initially, the main purpose was to identify the insect. So first thing was first step in any PMI estimation is to know what species we have. And during my PhD, we did a study on different groups of blowflies. So we started at the family level. As you can see, we had done on the Californidae, which is blowfly family. Then we studied on Chrysomyini, which is subfamily of blowflies. And then we did a study on Chrysomia, which is a genus of blowfly. When we started, we did not know much about these flies and their evolution. As you can know, for Chrysomia, basically everything we knew before I started working was wrong in terms of evolution. So our study solved lots of unknowns from these groups of flies, especially like in Chrysomyini, as you can see. Initially, we had two tribes, and they were, there was no phylogeny. So we did not know if they are monophyletic or paraphyletic. So I think initially I did on phylogeny, but then I thought, okay, this is important because when we have the database, we have a database to identify this fly, then it's easy to identify because we have seen when we did the crustacean identification, sometimes we felt, oh, it's hard. And when we started insects, it was harder because they are so tiny and many morphological characters are just those bristles on the body, which sometimes they have 3, 2, and many other characteristics. Those are small characteristics and it's hard to identify. And many times there is no way, if you have a maggot or pupa, there is no way to identify insect because sometimes we don't have a morphological key to identify. And without identification, you cannot tell how old that insect is because different species develop by different rate. So we did that. We also did some on Indian flies. So we had a collaborator from Punjab and we did develop a DNA based ID for different species of blowflies from India. So then the other way you can use insect to tell how old the dead body is to see the insect succession, because as the body decomposes, different group of flies comes at different time. So initially fresh, you get blowflies and fresh flies, as you can see on the screen. When the body gets bloated, mostly you see other kinds of flies and some beetles. When the body is in decomposition stage, decay stage, then you get a different kind of carrion beetles and other flies. And finally, when you have a dry or skeletonized stage, then you get other flies like black soldier fly and beetles. So these types of insect that changes can be utilized to tell how old the body is. So initially that was a common method, but in 2001, Dr. Bhass from Oak Ridge National Laboratory thought, okay, so when the insect changes, we know and we can tell how old the dead body is. Why not we can use bacteria to tell how old the dead body is? Because bacteria also will change, and there's an advantage of bacteria because sometimes people can remove bugs from the body and then we don't have evidence. For bacteria, it will be harder to remove. They will not see what bacteria are. So he started and he did experiment for almost 3 years, 3 months, sorry. And then he gave up because when he started culturing all those bacteria, he said, okay, we have too many. It's like we have everything except some bacteria that lives in deep sea vents. And then there is no way to identify these bacteria or microbes from decomposition process because you cannot keep track of how many are there. And then he did not have technology. In 2005 onward, we start getting high throughput sequencing, right? So 454 pyrosequencing, then ion torrent, then Illumina. Now we have another one. So there are many high throughput sequencing technology. The arrival of this technology helps scientists to identify all microbes from any environment, almost all, not all, but almost all microbes from any environment in a small period of time. And that changed the way we think about microbial ecology and microbiome. Then Human Microbiome Project started. And in 2010, we started using this technology. So as in 2011, we started working on human decomposition and how we can use bacteria associated with human remains for post-mortem interval estimation. So as you can see, this is the first experiment we did for the human decomposition. As you can see, we had 6 human remains and we put a pig as a parallel to see how they are compared to human. And then we put 3 under the cage and which were like having the insect exclusion cage. That means, as you can see inside the gray one, the insects were not allowed. So body was put inside, but there was no insect allowed, and vertebrate exclusion cage were there. And then here you can see we had insect access. So any insect can go and do. And then we had these sample collections. So we collected the buccal sample, skin, anal sample, soil under the body, and soil 1 meter away. As you can see with this one, we did a study because it was hard to get 2 human remains at one time. So we worked at a body farm in Texas, and the problem was we wanted replication. But then for that, we should have 2 persons dying at the same time, and both of them should have signed for donation. So it was hard to get samples to work on these replicated studies. So we got it, but it took 2-3 years to get those 6 remains together. So we had 2 at one time, 2 another time, and 2 another time. So we did not put 6 at one time. We had a 3 trial. And in this one, so we collected all these samples every 8 hours for 5 days. And as you can see here, what we observe is there was clear-cut difference between sample that had insect access and exclusion. So the green is insect access and blue is exclusion. Similarly, the sample that we collected under the body was very different from other samples. There was significant difference between 3 sites also, like anal, buccal, and skin samples were significantly different. But there was no difference between 1 meter and under the body, because I think it takes time to get soil saturated and soil biological community change. And we did only for 5 days. So during the first 5 days, the soil microbial community did not change much because of decomposition. So we did not see that change here, but we have clear-cut separation between the excess and exclusion. At that time, we also observed that there was two groups, one 0 to 3 days and another 4 to 5 days. As you can see, the bacterial composition 0 to 3 days were significantly different from bacterial composition that we observed in day 4 to 5. And most of them were insect-associated bacteria, like Ignacinaria, like Upartimonas. These are the bacteria commonly seen in insects, Acinetobacter. So these bacteria were common. And then we thought, okay, so if the insect, arrives, they brings bacteria, and that also change the bacterial composition of human body. And we can utilize these for PMI prediction. But the problem was we did a study only for 5 days. So we did not know what will happen after that 5 days. So that time, because it was first study anywhere in the world on microbial succession, we were the first to study that. And we did not know that, this decomposition is so slow and it will take longer than 5 days to get all the information. So next, we did another study, but this time we wanted to make sure we have a good replication. So instead of human remains, we used pig as a model. So we used 12 pig, as you can see here in the field. 6 had insect excess and 6 had no insect excess. We also wanted to make sure they are placed very far apart, so more than 20 meters away, so that there is no contamination from each body. And as you can see, these are the decomposition of 6 pigs that has insect excess. So it's almost, we placed those pigs and kept them for almost 60 days. And mostly I talk about ADD, which is time and temperature together, because accumulated degree days we use for time and temperature together. So as you can see, it was almost 1700 ADD and 60 days. The ADD we use because if we have ADD, we can compare with other study. With 60 days, it may be same, it may not be same for our area compared to some other area, because like in India, we have higher temperature. So in 60 days, we'll have higher ADD compared to what we have here. But the temperature is the important factor that determines how decomposition proceed. So we use ADD for most of the PMI calculation. Then we thought, okay, let's see how the body decompose. So initially we calculated body score. We call it total body score. That depends on how body, different head, trunk, and legs. They are different coloring, different decomposition process, and we use those processes to estimate PMI. As you can see in the left, we have log scale, and the R square value is really high to predict how old the dead body is, because the degree day can be converted into days, and we can tell how old the dead body is. So if we go to crime scene, if we see the body, we can score the body, and then we can tell how old the dead body is. The problem with this method, the total body score, is it will be good only till the decomposition is continuing. After a certain period, the body don't change much. You see just bone and skin, bones and skin, nothing is changing. Then how to use it? And that's when the problem starts. So like in this case, only till 900 ADD, we can use this method. After that, we cannot, because nothing is changing. So a score is like a highest score reached, and then we don't have a way. So then we started getting the bacteria and eukaryotes. So basically, I'm not talking about the methods here because we don't have that much time, but if anybody have question, I can talk it after the presentation. So we did 16S rDNA, next generation sequencing, MySeq sequencing, and then we did 18S rDNA for eukaryotes. So as you can see in both bacteria and eukaryote, on both sides you can see there is significant difference between different sampling time. As you can see, the initial time is 0 day to 61 days or 1700 ADD, and it's changing with time. There was significant difference between bacterial and eukaryotic structure with time or ADD. So we thought, okay, we can use that if we know the indicator groups. As you can see, in the bacteria, we found these bacteria, Clostridium, these bacteria were important group, actinomycetes, lactobacillus, and all those indicator group. Similarly, in eukaryote, we have different groups, mostly fungi, that were important. Like if you see here, in the eukaryotic side, we had initially we had lots of Ascomycota, then we had arthropoda, that's normally blowflies and things, and then we had nematode bloom. So you can see the pink one in the center, like from day 9 to 15, all were nematode blooms. So nematodes were eating on the bacteria and other things. And then when body start getting skeletonized, then mostly we had different fungal population. Like, if you see the Dapodi, Deboromycetaceae and other fungal population. So these groups changes with time, as you can see. And we thought if we develop a statistical model, then we can predict how old the dead body is. And that's what we use. So here we use a random forest model to predict how old the dead body is. And as you can see on the top, these are the indicator group. So the highest was Diptera and then other fungal group. And Rhaptidia is a nematode. So those were important group. And then on the right side, it just shows how they change with time. So there are different groups, at least top 5, and then how their abundance changes with time. And then on the bottom, at the bottom, what you see is on the y-axis, we have predicted ADD or PMI, and at the bottom, we have actual ADD. And then this is a fit line where we see that we can estimate PMI for these remains with an error, random mean square error, RMSE, of 177 ADD, which is equivalent to 6 days. So we can estimate PMI based on eukaryotic data by 6 days of error. So that will be maximum error of 6 days plus and minus for 2-month-old dead body. Then we thought, okay, that's good, but what about soil? So when the body decomposes, the material goes down, right? How long it impact the soil? Is the soil going to recover, or it will remain like that? What kind of bacteria changes when the body decomposes and all the liquid goes down? So initially what we did, we had 11 human cadavers, and those human remains were 3 days to 714 days, almost 2-year-old. So we collected sample at 0 meter, that means just under the body, then at 1 meter and at 5 meter. We wanted to see two things. First, is there any change in soil microbiome compared to a control soil at 5 meter? And then if yes, then how long this impact goes? Is it just under the body, or is it up to 1 meter from the body? And to do that, we did 16S rDNA sequencing. And as you can see here, there was significant difference between 0 meter sample compared to 1 meter and 5 meter sample. Also, there's a spread of samples at 0 meter. We said more beta diversity for sample under the body. So the human remains also have impact on microbial composition. And at the bottom, what you see is when under the body, that is 0 meter, we have less acidobacteria compared to other samples. So if you see the acidobacteria, so acidobacteria is common in soil sample, but when we had human remains, the acidobacteria population declined, and actinobacteria, gamma proteobacteria, and bacilli population increased in soil under the remains. So these groups were significantly high under the remains. We also observed that the relationship between bacteria and ADD was not good for PMI prediction. But when we use the cumulative precipitation, because Texas is kind of a little bit dry, so water had more impact than the temperature, and we got a linear relationship between actinobacteria abundance, and cumulative precipitation. We also observed that the cadaver starting mass, then heavier the cadaver is, more gamma proteobacteria we had. So there was a strong relationship between cadaver starting mass and gamma proteobacteria. So these two things we observe. So we observe that we can use this information to tell where the dead body is. Now this is very important. If you are looking for dead body and you have samples, we can identify human cadaver decomposition islands. Or if we sometimes the body is moved, but suspect particular site, then you can use this information to tell whether there was any body decomposing or not. We did not test how this will change if we have a human body compared. Right now we had human body. What will happen if we have a pig or dog or deer? Will they decompose the same way, and will they change the community the same way? But at least we can tell that this site had some kind of decomposition going on based on the microbial community composition. Next, we wanted to see, okay, that was on the land. What will happen in water? So what will happen in water if we start, so we did experiment in 2 water systems. And this is important because, I remember as an undergrad, Dr. Patange sir, he taught us aquatic microbiology. And we had very limited information on microbial composition of different water bodies. And what we did not know much. Because of high throughput sequencing, now we know a lot more than what we knew 20 years ago. So basically, we did experiment in Virginia in 2 water systems. On the left is a lake, Henley Lake, it's a lentic system, and then here is a lotic system, which is James River. So this river is, the experiment site we call Rice River Center, which is owned by our university. So we can use boat, we can put the dead remains, and we had a permission to do experiment. But initially we thought, okay, mostly what happens when somebody get drowned, mostly we get bones, right? So how we can use bone to tell how old the dead body is if we get body after years, maybe 1 year or 2 years or something like that. So we put two bone type, scapulae and rib. So we use this because when body get drowned, these 2 bones are intact for longer period of time. And also it was easy to make replication because we can have all, replicated experiment. So we did this one for almost 2 years, almost 1.5 to 2 years. And for each we made like a dog cage where we put 5 rib and 5 scapula, and then we put each one in the lake and river, and then exactly after 250 ADD. So this ADD time was sometimes, you have only 10 days, sometimes you have a 25 days. It depends on the temperature and season. But then it was recorded online. So at live, we knew that when it will reach 250 ADD, and then we went and collect sample. So as you can see in the first graph, this shows bacterial composition in rib and scapula under lake and river environment. What you can see here is the initial composition. This one is initial composition for all the sample, like at 0, the bacterial composition is same in both rib and scapula, almost similar. But when we put them in the water, then the composition changed significantly. As you can see in the rib bones, in both lakes and river, mostly Clostridiaceae and Ruminococcaceae was more dominant taxa, and there were other taxa also that changed. In case of scapula, it's a Holophagaceae that was more common, and that changed a lot. Other Clostridiaceae was also there, but that declined after initial period. And here we see in case of lake, we have Spirochaetaceae that change a lot, and the Holophagaceae. So in the scapula, we had Holophagaceae, and in reef, we had Clostridiaceae that changed a lot. So I see clear-cut succession. We have lots of them. There were some, the black one you see, they were unclassified because still we don't know much about the aquatic system. So they were around, especially in the latter time point, I see lots of them not classified or did not get classification. So we did not identify. But what we did is when we used this one in multidimensional analysis, we saw that there's a clear-cut succession of bacterial community in both reef and scapula. What you see is the community at the lake was very different from river, and similarly in the reef also and scapula also, it changes with time. So the lighter the color, the more ADD they have. The deeper the color, the less ADD they have. Similarly, the circle is for the lake and triangle is for the river. So we can see the clear succession with time for these samples. On the right side, what you see is the water sample. So with the bone, there was not that much change in the water sample in both river and lake. As you can see at the class and family level, the water composition of the bacteria did not change much with time in both river and lake. So although the bacterial composition of bones changed, I think it did not impact that much to surrounding water. I don't know how it will change if we have a real human remains or pig with muscles and tissue and things like that. Because right now it was bone, maybe it did not impact that much to the surrounding water. But once we'll have the soft tissue, it may have impact to the water, and there may be, interaction between water and the tissue as well as tissue and the water. So both way, the bacterial composition may get impacted. Then we use all this information to develop a model for prediction. Again, what will happen, because right now we don't know much in water. We don't have a way to tell how old that body is if we get a body from water. So this will be the first kind of a study where we did on bone. We don't have any microbial method or any other method except they say total aquatic decomposition score, which is kind of subjective and it changes with person to person. So what we observe here again in the lake and river, these are the groups of taxa that are important for PMSI indicator. So PMSI is post-mortem submersion interval, how long the body was in the water. As you can see, it's in the reef, it's like mostly depending on the river or lake. In case of lake in reef, Synthropomonadesi, the pink one, is really the most influential predictor taxa. But in scapula, it's different taxa. In river, we have slightly different taxa, but it's all together we use, then we can predict the PMSI. So as you can see here, so we in the reef and scapula, in case of lake, scapula was better predictor. We were able to predict 94% of variation in ADD using these models. And as you can see, in case of lake, scapula samples were able to predict PMSI with an error of 37 days out of almost 2 years, 1.5 years or something. So you can get error of 1 month approximately by using this method in either lake or river. So it's not perfect, but it's better than knowing nothing about the samples that we get. So we can tell, okay, at least we can tell that how old the bones are in particular aquatic system. Then we wanted to know what about the eukaryotes. So in the water, we collected bacteria. We also collected eukaryotes. Sorry, I think there is error in the slide. This should be eukaryotic succession. It's not bacterial succession. So it's a eukaryotic succession. So what we observe is, as you can see on the right, the blue is water and green and red is scapula and reef. As you can see, the water samples have a different eukaryotic composition. They make a different cluster compared to scapula and water. These two are together. And then scapula and water did not change much. And when you see in case of water, again, the eukaryotic composition did not change much. But in terms of rib and scapula, they had similar composition and they changed with time, although we did not get, taxonomy at lower level. So it's like a phylum, class, family. We don't go below that because the eukaryotic classification is very complicated and challenging. We get lots of unclassified. Sometimes they say, okay, this family, they will say arthropoda, but nothing below that. Sometimes they say, particular family like nematoda, but we don't know what happens after that. So some of the taxonomy problems are there, but what we see is there is a clear-cut changes. So the one that is a Pyronosporomycetes, which is like a water mole. Initially we have water mole, then it declines, and again it comes. Now we don't know what kind of water moles are initially and what kind of water moles are at the later time of decomposition. Same thing in both bone types. Then we have lots of unclassified group that we don't know. We also have, lots of Celiophora and other groups that is changing with time. So we use this information to develop again another model. And with this model, for the same sample we use for bacteria, we were able to do prediction of PMSI with error of 104 days for rib and 63 days for scapula. And this is for the leg sample. We did not analyze river sample yet for eukaryotes, but our plan is to combine these data. How about we combine both eukaryote and bacteria and develop a model using combined data for both river and lake, like a universal model where you can use it for any water body. And then we can say, okay, this may be the error. So that's the work we are still doing right now. We are currently doing. This was a work by a PhD student. She did all these works and a master student. Then the third thing we wanted to know, as I said earlier, there's an interaction between insect and microbes, right? Because when the body decomposes, insect come and it brings the microbes. There are lots of microbes that are specifically from insects. So we wanted to know what microbes are there in insect and how they influence this interaction, right? So we used 3 flies, black soldier fly, which is generally common at late decomposition period, and then blowflies, which is common during early decomposition. And what we have observed and what we have seen is, and that's also important for the fisheries point of view, because these black soldier fly larvae can be used for animal food. Right now it's pretty common. Like the lab where I was working, they have started a company, and because this theme is entrepreneurship, I think there is a huge opportunity for people to get involved in this culture and production of larvae for the animal food production. And this the company that I'm talking is from my postdoc mentor. So my postdoc mentor opened this company, I think 2 or 3 years ago, and they named it Pop Worms, where they do all the hygienic testing and quality control and everything, and making sure they use the material that is recyclable, so they don't have to buy the food. So recyclable material, and that get converted to larvae, which has 43% protein, and they use it for mostly for poultry industry, but they're also working for aquaculture. I don't know much about nutritional value and how much useful it is in terms of aquaculture, but they are doing some experiment to make sure they know all the quality things for these larvae. But I think that's one where they have started and they are getting good income from just selling the larvae in a particular scientific way. So this black soldier fly, what we did is we wanted to see the microbial composition of these flies. Are there any, pathogenic bacteria and other things that will impact, whole, poultry industry and other things? And what we observe here is, as you can see, the black soldier fly on the right side have very different microbial composition compared to blowfly, which is Lucilia cuprina and Lucilia sericata here. As you can see, the blowflies have mostly, they have lactobacillus or lactococcus, but in terms of black soldier fly, they have many other bacteria, but Providencia and Bacteroidetes were the two most dominant bacteria. And these were very significant. The microbial composition between these bacteria were very significant. We also did all the larval stage. I did not include those results here. We did egg, larvae, pupa, adult, and then we tried to, test how these, how the bacterial transmission happens horizontally and vertically. We wanted to see how many bacteria are unique between these three groups. As you can see, the black soldier fly, and they eat all this waste material. So automatically they have more diversity compared to the blowfly. Although blowfly also eat, decomposing material, but I think black soldier fly eat lots of other material that blowfly don't eat, like organic material that is not of meat origin. So what we saw is, in all these flies, the four bacteria were common: Providencia, Proteus, Ignacinaria, and Lactobacillus. These four were common among all these flies. And then there were unique bacteria in each species. What other things we noticed is, we also got from this egg, we extracted bacterial composition, mixture of bacteria, and we found that this mixture attract blowfly and black soldier fly oviposition. That means if you know the bacterial composition, you can manipulate the flies for oviposition and increase your production, capacity, and how to utilize those resources. And when we sequence all those mixture that we got from the egg, we found that these are the 5 groups that were common in those mixtures that attracted flies more for oviposition. So this information about microbes can be utilized for several purposes. As I said, it can be used for oviposition preference. It can be also used for where you want this fly to lay egg and how much resource will be utilized for this method, and then rate of decomposition. Okay, so that we're on the dead remains. Now I will move towards living person. Okay, so it's mostly on human microbiome and application to forensics, but the technique that we develop can be used for any macrobiology work for any subject. So what we did is, initially, we wanted to know, okay, we know the human microbiome, how we can use for forensic. And the one thing that came to my mind was body fluid ID. So here in US, for any crime lab, the most important task is to make sure they know what biological sample it belongs to. Is it feces, it is saliva, it is semen or anything. And it's very important because sometimes you don't want to do DNA typing and everything if it's not important, if it's not body fluid, or if it's not particular body fluid, right? Also, sometimes you want to make sure you know which body fluid it is, because sometimes the DNA is from particular person, but that does not tell how the crime happened. Maybe if you know that the DNA came from saliva, then it will say something else. If we know the DNA came from semen, then it will have different meaning. So it's important not only to know from where the particular sample came, but also to know what part of the person donated that sample, or the sample came from which body fluid. So it's very important to determine body fluid ID. Unfortunately, we don't have a method that is confirmatory except macroscopic examination of sperms. So far, up to now, all the methods are either presumptive or they have some other problems, but none of them are confirmatory. There are some exceptions, some false positive, false negatives. So we don't have a method. So I started thinking, okay, when we see the microbiome, we have several advantages. And these are the challenges we see in the body fluid. For example, as I said, we don't have any confirmatory methods. Some method consume samples. So when you test like immunological testing or any, serological testing, you consume sample. So every time you have to do for, okay, you suspect it's semen, you tried it semen, it did not work. Then you try saliva, it did not work. Every time you do one test, you consume sample and you don't have that much evidence. So we need to have a method where one test can tell whatever sample it is. We don't have to use several tests, right? When we do the messenger RNA based method, then it's a problem is we have to do separate primer, separate consumption for each gene that we want to amplify. So it's kind of expensive and time consuming also. And basically we don't have a proper method yet. So we started thinking about that. But before that, we did not know how to know how much bacterial DNA we have, because even the human microbiome project did not quantify. They just use the total DNA. But when we extract DNA, we don't have only DNA from human. We have DNA from bacteria. We have DNA from fungi, right? So we did not know how much is the microbial DNA. So first thing we did was to develop a method, because when we started, we did not have a method for qPCR analysis and identify bacterial DNA. So we developed this method, and that was published as a cover page on this journal. And what you see here is different body fluid and how much is the human DNA and how much is the bacterial DNA. So the red is bacterial DNA and green is human DNA. As you can see, the feces and urine have more bacterial DNA than human DNA. And that's expected, right? It's a waste material. In feces, more than I think 90% is just bacterial DNA. As you can see on the left side, semen have almost very low quantity of bacterial DNA. It's all human DNA. So its ratio is 1 to 6,000. But in other body fluid, like in feces, 37 times more bacteria is there than the human DNA. So it's a lot. In urine, it's 8-to-1 ratio, right? But in other like saliva, it's 1 to 50, but still it's a lot of bacteria that can be used for identification purpose. So we did human DNA from same sample and bacterial DNA, developed the method, and now we know that how much bacterial DNA we have. Other problem with forensic, and it may be quality control criteria for other things also, is to make sure we know how much DNA input we have, either human or bacterial. You cannot go without telling the DNA quantity if you go for STR typing or DNA typing. So it was very important for quality purpose to know that exactly how much DNA we are adding, bacterial DNA, and what is the minimum that can give the same result and without any problem. So we did this, and this can be used for the method that we developed, can be used for any sample. It's not a forensic sample. Any sample can use that same primer to quantify bacterial DNA, any types of sample. So what you see here is like initially we did a small number of samples, 5 individual, and we wanted to see what impact different concentration have. So as you can see in this blue is saliva, black is feces, green is urine sample, and red is menstrual secretion, and yellow, orange is vaginal secretion. The shape, the larger the shape, the more DNA we added, like 0.3 nanogram DNA we added. Second was 0.07. Third was 0.02 nanogram, which is 2.02 nanogram DNA. So we tested 3 concentration. As you can see, sometimes the concentration we used did not impact, but there is a difference between individuals. Sometimes different individuals have different, bacterial composition. And also each shape is one individual. So some sample, we had all individual having similar bacterial composition, but like in vaginal secretion, we had differences between people. Similarly, in urine, we had difference between male and female. So we want to test what is the minimum bacterial DNA that we must have to make sure it works every time our result is consistent. So we observed that, as you can see in this figure, these are different body fluid. And as you can see the concentration and these are the amount of DNA we used. We started from 0.3 and we go down to 0.001, which is 1 picogram. And what we observe is in most of the sample, we had a consistent result till 5 picogram. But the last one, the 1 picogram, gave result that were not common. So that was the problem, because then we recommended that, okay, any bacterial method you use, for that, you need to have, at least for body fluid ID, have at least 5 picogram of bacterial DNA to go for. Then we did this body fluid ID from lots of samples. So right now we collected almost 1100 samples from different people. As you can see, we had people from different age group, like less than 18 to 30, 31 to 50. And because it's university campus, the number is more towards 18 to 30, because we had lots of students donating the sample. And we had more Caucasian and African American people donating the sample than Asian, but we had quite a few Indian and like a Chinese descent people also. We had many mixed race people, and as you can see, we had almost 200 samples from many body fluid for this purpose. And then first thing we had a problem is the reagent blank. For every extraction, we use reagent blank, and reagent blank had some sequence. Now we were very confused why we are getting reagent blank. It should be 0, right? Because it's a control. Then we talked to Kaizen about the problem, because we were getting some bacteria in all reagent blank. And then they said that they don't certify that it will have no bacteria. So the DNA extraction kit is not 100% bacteria free. So, and mostly the bacteria that we see in those kits are water-related bacteria like Burkholderia, Acinetobacter, Colobacter, and like that. But mostly Burkholderia is lot in water, and the kits also have that. So they did not purify several times to get these bacteria not there. And then when we do this high-sensitive technology and sequencing, then we find these things. So when we did analysis, we subtracted these bacteria from all the sample to make sure the bacteria we have is only from those body fluid samples. And as you can see here, this showing the diversity in different body fluid. And as you can see, the blood, feces, and semen, although semen had less bacterial DNA, it was much more diverse than other body fluid. Then we see, as you can see in this figure on the left, heat map, there are different bacteria in different groups. Don't worry for name, but as you can see the color, these groups of bacteria are very distinct in particular body fluid. When we did the analysis of molecular variants, we observed that all body fluid had distinct bacterial composition except menstrual blood, vaginal secretion, and urine from female. Then we developed several machine learning methods for this purpose. So we use the support vector machine, linear classifier, then SVM, radial classification, decision tree, random forest, XGBoost, and all these 5 gave the accuracy in the range of 88 to 92%. Because SVM linear was the best method, we choose that for validation purpose. So what we observed is when we use this method, we were able to predict body fluid with 92% accuracy. As you can see, they are different. We use the precision, then sensitivity, recall, and F1 score, which is a geometric average of precision and recall. So as you can see, for some body fluid, we had very good success, like 100% for blood, feces, saliva, semen, and then we had less success for urine and female urogenital tract samples, like a menstrual blood and vaginal secretion. We were not able to separate because they had mostly lactobacillus. And also in urine, we had mostly lactobacillus. So these three samples, female urine, vaginal secretion, and menstrual secretion, cannot be differentiated using this method. But if we combine them, then yes, we can separate all other body fluid with high accuracy. And this, in the blind validation, the study was done by someone else. So they did not know what sample they are doing. And I used our model to predict the body fluid. So overall, what I talked today is what are the things I am doing and how these techniques can be used for any science, forensic science, fisheries science, biology, any other subject. Mostly we have done work on evolution of blowflies, DNA-based identification of flies, microbial decomposition process, human microbiome and its application, and interaction between arthropods and bacteria. With this, I would like to thank all the funding agencies. I received money from BCU, National Institute of Justice. The body fluid ID grant was funded by National Institute of Justice. Some of the validation study is funded by National Science Foundation. We got some money through different PI from NIH, and then we got several funding from American Academy of Forensic Science, which is the largest forensic organization in the world. So they supported some of the funding for this work. And I would like to thank all the members of my lab. There are many collaborators. I did not talk, but the study was done with several collaborators: Tal Simmons, Dr. Sarah Cecil Williams, and many other collaborators. These are the master and PhD students in my lab, and I will be happy to open. I will be happy to have any questions. Sorry, I think I took more time than I had, but thank you, everyone.
Thank you, Dr. Baneshwar, for a very fantastic lecture on the bugs and bacteria of interest in forensic and fisheries science. So many students, especially from the microbiology department, they might be motivated because of this particular presentation. How exactly we need to carry out the research work, how to come to the conclusion so that our results will be beneficial to the overall society. Let it be fisheries or it may be a forensic science.
Yeah, I think we have to. Sorry,
yes.
Go ahead.
Yes, Baneshwarji.
Yeah, I was just saying that this microbiology or bacteria and this it's everywhere. So the microbiome work can be used for any discipline, and in fisheries it's more vital.
Yes. Thank you, Baneshwarji. I think we have not received any question in the chat, either at Zoom. In Zoom, we have about 60 participants. And in case of YouTube streaming, we have about more than 100 participants. So right now, we are not having any question in this one.
Dr. Chaudhary.
Yes.
Dr. Chaudhary, please.
Yes.
Dr. Chaudhary, sir, I have one question with Dr. Baneshwar. Can I ask him a question?
Okay, ask.
Okay.
Hello, Dr. Baneshwar.
Yeah, hello.
Hello, Dr. Nitin Samadia. Very nice to see you and listen to you after a very, very long break.
Yeah, thank you, sir. I think it's nice
talking from our place now, but very happy to see you here.
Thank you.
I am working here in Sindhudurg district of Maharashtra on one fisheries project. You know that project.
Okay.
We are here working on live food production of black soldier fly.
Wow.
Last year we have taken up this project over here and we are working on that.
Yeah.
So fortunately, we have got one local species of black soldier fly here itself, and
wow.
So the potential of the same species we are exploring over here. So my request and my question to you is that how we can collaborate this work. Is it possible that we can collaborate this work with your organization, as you are working in a very reputed university of USA?
Yes, sir. I'm open for any collaboration or anything you need, but I can also connect you to the
Okay.
other people who is
Okay.
involved in the entrepreneurship, like who has started a company or who have already successfully running the company.
Okay.
So
Okay.
if you want, I can connect to those, like my postdoc mentor. He is
Okay.
like a managing director of that company also. So
Okay.
he
Okay.
do all this business, and my colleagues have done whole genome sequencing on this flight. So we have all the people working, and I will be happy to either help if it's in my expertise or to connect you to the people who have the expertise in that area. I know some people in like my postdoc mentor, they had a good collaboration, a company that makes M&M and all candies, Mars Corporation. They are spending lot of money on utilizing black soldier fly larvae for animal food, and they are already lots of work going on. So yes, I think I will be happy to help and connect to people who will be helpful. But that's really nice to know that you have started. This has a huge potential.
Yeah, yeah, definitely, definitely.
Thank
And
you, Dr.
we
Baneshwar.
got a local species over.
And that's a great. We can know, we can do lots of things about that. We don't know much about that local species.
Yes, thank you, Dr. Baneshwar. There is
Yeah,
one question in YouTube.
yeah,
Mr. Sagar Shinde here has asked that,
yeah.
what if a dead body found in water is there, same process and the test to determine PMI?
So what if dead body is in the water and then?
And then whether the test and process is similar, he's asking.
Yeah, so the question is the decomposition process is similar, but water don't have the insect that is on the surface. So like in the terrestrial system, we had the blowflies colonizing the body. In water, when somebody get drowned, initially body goes down, then it comes on the surface. So when it comes on the surface, the blowfly colonize that body. But once they start eating the tissue and things, again it goes down. So when you recover the body, you can use insect. For the time being, it's on the surface because of bloating, the body comes out. But after the insect start eating, the body goes down again. So then if it's inside, then we can use the bacterial and eukaryotic composition to tell how old the dead body is. Now we have not done an all water body, so we don't know the method we develop will be useful for all system. Like we have to see broader scale, like we have not done on different geographical area to see if we get the similar result or not. But in this area, it was working fine.
Then one more question from Mr. Suraj Boyer.
Okay.
What are the major differences in decomposition of a fatty body and muscular thin body?
Okay, so I think that's what we talked little bit, but we don't know much. What we saw is the gamma proteobacteria is more when the fatty body decomposed. So the amount of gamma proteobacteria is higher and higher because we had several human remains with different weight, and most of the higher weight side had more fatty body. So we don't know exactly whether it was just coincidence because the number of bodies were very less. We did not do a study with many replications. And so we don't know if it's just because of high fat or because of some other reason. The person we had the more gamma proteobacteria and that increased the number. So we don't know exactly how that will impact the bacterial succession.
Though not related to our this one, but many people, since there is a COVID pandemic, there is one question related to that. Is there any case found COVID-19 infection in live fish or freshly caught fish? Dr. Ashwin Kumar is asking this question in YouTube.
Yeah, I'm not sure about I've seen in US they had dogs infected with COVID,
Okay.
but I don't know about fish.
Okay. 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 So thank you, Dr. Baneshwar Singh. I think we have
Thank you, sir.
covered.
Thank you, everyone. It was nice talking to you all, and
And
I will be here. So any question will come later. Then
later also. Yes, 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 yes. Thank you very much.
Yes.
I think our Vice Chancellor, sir, Honorable Dr. Sanjay Sawant, sir, and our uh, our guest of honor today, Dr. Jaina, sir, has also joined our webinar. So our pending inaugural function, I think we'll now held that, we'll organize this particular function now, and then we'll go for other two lectures. So thank you, Dr. Baneshwar Singh, for very nice presentation on the very different aspects. As the fishery students, all the fishery students, there are many fishery students, those who have joined this particular webinar, they must understand one thing. What we learn in fishery science is basically the fundamental thing related to the science. And this particular knowledge of the science can be useful to many different sciences. So
Yeah.
my guide and my very good teacher, very fond teacher, Dr. Sankoi sir, he used to say that fisheries is not one science. It is the amalgamation of all the sciences. And
Yeah.
it is true, considering this particular aspect, that you have shifted from fisheries to the forensic
Yes.
sciences.
Just I want to say a few things like, I did not shifted or move because of my interest.
Yeah.
I moved because it happened.
Okay.
I did not move like I want to move. I was so much. And then I was working on crustacean taxonomy. And then my PhD mentor said, I work on this fly, which is similar technique. And that's how we started. And then we getting more and more information. But one thing that I learned in undergrad and master, that was very helpful because the technique we learned, we use the same technique. So it did not take me so long to get adjusted with other discipline.
Yes. And then another aspect that Dr. Nitin Sawant has asked you the question regarding the BSF.
Yeah.
BSF larvae, they have started work since last 2-3 years. They are working on that. They are very successful in producing the larvae and also many things they are successful. So I think we'll have collaboration with respect to that particular aspect.
Yes,
So,
and for the BSF, I think my postdoc mentor was, I think he's a pioneer in that area. So if you see the paper, mostly it's from him and his group. And I think it will be really good collaboration for all of us. If anything is needed, I will be happy to help and collaborate.
yes.
Thank you, sir.
Yes. So thank you, Dr. Baneshwar Singh. Now, I think we'll move on to our inaugural function as our Dr. Jaina sir and our Vice Chancellor joined, also our directors, Director of Research and Director of Extension, Director of Instructions, all are there. So I think we'll go for our inaugural function. And so dear students and ex-students, alumni, all our stakeholders in the fisheries science. So I welcome all of you to the Foundation Day lecture series that is on the 39th Foundation Day of the College of Fisheries, Ratnagiri. Fisheries sector forms an important profession and economic backbone of the Konkan region. This region of Maharashtra is bestowed with vast natural resources with the potential to generate and support employment opportunities in various sectors of fisheries activities. Considering the need to develop the fisheries with the scientific approach, the government of Maharashtra established the first fisheries college at Ratnagiri under the erstwhile Konkan Krishi Vidyapeeth, that is Agriculture University, now known as Dr. Balasaheb Sawant Konkan Krishi Vidyapeeth. Inauguration of the College of Fisheries was held on the 4th of August 1981, and hence we are celebrating our Foundation Day on the 4th of August every year. On this occasion of the 39th Foundation Day, it has been decided by our college to organize every year the Foundation Day lecture series by inviting the pioneers in the field of fisheries to deliver the special lectures and provide an opportunity for the professionals in this particular field to interact and consolidate the foundation of fisheries science. This year, due to the COVID-19 pandemic, The Foundation Day lecture series is in the virtual mode by organizing a webinar on entrepreneurship development for sustainable fisheries. We are fortunate to have three towering overseas our students who are doing the wonders in their respective field. They are Dr. Baneshwar Singh, we have heard him. Then Dr. Amit More is working as assistant professor in Arabian University. Dr. Naga Murli, I think he is Mr. Murli, but he is now doing his PhD also. So we can say him Dr. Naga Murli. Dr. Naga Murli, he will deliver his lecture on the aquaculture. And the College of Fisheries Ratnagiri proudly presents the Foundation Day lecture series to inspire and motivate the students and the fisheries professionals by connecting them with the performance in the various fields, involving a rich spectrum of ideas and inspiring concepts. It brings an opportunity to interact with such a personalities who are at the realm of fisheries research today. For the inaugural address, we have none other than Dr. Jaya Krishna Jaina, popularly known as Dr. J.K. Jaina sir, our Deputy Director of General Fisheries Sciences from the Indian Council of Agriculture Research, New Delhi. Welcome, sir. And
thank you so much. Thank you so much.
Your blessings, your love to the College of Fisheries Ratnagiri and joining for this particular webinar.
Good afternoon.
I also welcome. Yes, sir.
Good afternoon, doctor, and welcome,
Yes,
welcome to this webinar and their function.
Thank you
welcome,
so
sir.
Thank you
I
so
also
much.
welcome our beloved Vice Chancellor, Dr. Sanjay Sawant, sir, who is our inspiration, who always keeps on us motivating for organizing different seminars, and who is also president of our today's inaugural function of the webinar on entrepreneurship development for sustainable fisheries. I also welcome our Director of Extension, Dr. Sanjay Bhave, sir, Director of Research, Dr. Parag Gardankar, sir, Director of Instruction, Dr. Satish Narkade, sir. I welcome, sir, all of you as a special guest for this particular webinar and also for this particular function. I also welcome all our head of departments of College of Fisheries Ratnagiri, Dr. Naik, head of department of aquaculture, Dr. Mohite sir, head of department of fish processing technology, Dr. Mrs. Swapnavajya Mohite madam, head of department of fisheries biology, Dr. Anil Pause is the head of department of fisheries hydrography department, Dr. B.R. Chavan sir, who is the head of department of fisheries engineering, myself working as a head of department of FRAC. I welcome all those head of department of College of Fisheries, all the academic staff members, very enthusiastic staff members for this particular Foundation Day lecture series, all our ministerial staff, our students, our ex-students, our alumni, and all our stakeholders, fishers, fishermen, fisherwomen, for this particular Foundation Day webinar on the entrepreneurship development for sustainable fisheries. So this year Foundation Day lecture, first lecture, already Dr. Baneshwar Singh, he has delivered, who is working as the Associate Professor of the Forensic Science in the Virginia University. Then the second lecture will be by Dr. Naga Murli. He is the Managing Director of the Voltas Brim Limited, Ghana, his own company. He will be talking to us on the trends and techniques of tilapia farming for successful entrepreneurship. And our third lecture will be delivered by Dr. Amit More, who is working as an assistant professor in the Arabian University, USA, and who is also founder and president of Idea Food Industries Consultants. So he'll be delivering his lecture on preparing your career in the futuristic seafood industry. So with this little long and introduction and welcome function, I request our beloved Dean and Associate Dean of College of Fisheries Ratnagiri, Dr. Prakash Shingare sir, to give the preface to the webinar. Dr. Shingare sir.
Good afternoon to everybody. Distinguished guest and today's chief guest and guest of honor, beloved Deputy Director General Fisheries Science of ICR, New Delhi, Dr. J.K. Jena sir, our Honorable High Chancellor, Dr. Sanjay Ji Sawant sir, who is always inspiring us to organize such webinars and functions to enhance and consolidate communication with present and past students. Our respected all directors, Director of Extension Education, Dr. Bhave sir, Director of Research, Dr. Hadankar sir, and our Director of Instruction, Dr. Narkade sir. Proud parents, even prouder Kofians, all heads of department of the Faculty of Fisheries, all academic staff members, ministerial staff, and dear students and ex-students. I welcome all of you to our 39th Foundation Day. Sir, allow me to introduce myself. I am an old boy who has become the new Associate Dean of this Faculty of Fisheries. I said old boy because this is the college where I completed my graduation, post-graduation and doctorate. And after education, I served this university for the last 27, 28 years. And now bestowed with me to lead this organization. I know it is a rare opportunity, and this rare opportunity is given to me by our Honorable Vice Chancellor sir, Dr. Sawant, Dr. Sanjay Ji Sawant sir, who is also the alumni of our university. Sir, I say that not so much to inform you, but as to remind myself of the honor you have bestowed on me by entrusting to me and to my colleagues in the Faculty of Fisheries to care the education of the children. You have, sir, reminded us that teaching is a noble vocation known to be the civilized world. The College of Fisheries Ratnagiri is a unique college for the Fisheries Educational Institute. Its goal is not only to produce fisheries graduate and postgraduate students, but also to produce the true and honest citizens of this country who will serve fisheries community not only in the Konkan region, not only in the Maharashtra, not only the nation, but also international level. Our students have performed extremely well at all levels, that is state, national and international. And today we have such three distinguished and highly successful students who are working at the international level, who are the speaker of today's 39th Foundation Day. Dr. Baneshwar Singh, Dr. Amit More, and Dr. Nagamurli. Formal introduction of the speaker will be given by the Dr. Ketan Chaudhary. But I must say that these are the proud students of this college, and we all teachers are very happy today to listen to them on 39th Foundation Day. Sir, as you know, as a constituent institute of Dr. Balasaheb Sawant Agriculture University, the institute has three mandates, that is teaching, research and extension education. In all these three sectors, college have performed extremely well and to satisfaction of our university and by the ICR. Our college is established in 1981 at Ratnagiri, which is the first college in Maharashtra, and that was the third college in national level. Our college is under the Dr. Balasaheb Sawant Konkan Agriculture University, Dapoli, and in the span of last 38 years, the College of Fisheries has significantly contributed to the development of state as well as the Indian fisheries. Sir, so far more than 1,000 students of the BSKKV has been produced by this college under the Dr. Balasaheb Sawant, Konkan Agricultural University. Then more than 325 MFSC students and 20 PhD students have obtained the degree from this college, who belong not only to the Maharashtra, but also to the different states of the country, from the Jammu Kashmir to West Coast up to the Kerala, and on the south East Coast, that is from West Bengal to the Tamil Nadu. So all over the country, students have participated, taken the admission in this college. This important human resources of the state has been shouldering the responsibility of administrator, entrepreneurship, then employers, academicians, technicians, then managers, fish farmers, hatchery operators, fish exporters, quality control inspectors, bankers, aquarists, with a professional dedication in their field. So many fields, not in the fisheries field, but also other fields also, our students are working at the high position. Now, as a time is the limit, I will just give the important milestones of our institutes, how our institute has developed since 1981. Sir, our institute has established in 4th August 1981 in a small building, which was the research station called Marine Biological Research Station, which was established in 1958. And in that building, our first batches, that is first batch of the 1981, has been started in the same building. And at that time, the course was the 3-year course duration and only 20 intake capacity. But after 3 years, the ICR started nominating these students or inclusion of the students in the degree and PG level in our institute. Then in 1991, this 3-year degree course, it has been converted into the 4-year degree course as per the ICR instructions. And in 1996, that is after 5 years, our university and faculty adopted the ICR modern syllabus for the BSc students, and also the intake capacity of the degree level was increased from 20 to 40 students. And simultaneously, the postgraduate MFSc course by course and partially by research was started in 1996. Till that, all students were doing the MSc and PhD purely by research. Then in 2010, the 14 committee syllabus was formed, and BASMA committee has adopted the syllabus of the BASMA committee. Then in 2017, the 14 committee was syllabus of the 14 committee was adopted by our institute. And then the different courses in the PG level, that is MSc and PhD level, were also offered. In MSc level, the 9 disciplines, and for the PhD level, 6 disciplines were offered for the students. Herewith, we have also have the 20 number of the PhD students have taken the degree, and out of that, some of the students, they have taken their degree in foreign countries. Our college have started the ARIS cell, that is Agricultural Research Information Center cell also was started. Then our university has given the facilities to the faculty, like recent fishing research vessel for the onboard in the to use for the students for marine fisheries. Then also the different laboratories in the different departments were well established under the Dr. Balasaheb Sawant Agricultural University in the Faculty of Fisheries. Then our college has started the digital library, which is the recent development in our faculty, that students are getting the digital library, online library. Then considering the importance of fisheries in the state and Indian economy, along with the scope of expansion, government of Maharashtra, they have initiated the research work in the Konkan region, specifically the 2 research stations, that is first research station, that is Tarapurwala Marine Biological Research Station under the Faculty of Fisheries. It was actually established in 1951, but when our university was formed, then it was transferred to the university in 1971. another research station at Ratnagiri, that is Marine Biological Research Station. It was also transferred to our university in 1971. And when university was established, the university also established the new research station, that is Harland Research Station, Panvel, and also the Fisheries Research Center at Molde. These two research stations were established by our university for the development of this fisheries faculty. This research station has also played a very major role, that is considering the various research program to cater the need of the farmers and fishermen community. The fish production unit, that is the circular hatchery, it was established at the Mulde Research Station. Then Sibas Research Station, Sibas Research Activity was established at the Panvel Research Station, and our Mumbai Research Station. Since long that the research station is working on so many aspects, and the research station has given the recommendations to the not only to the farming community but also the state government and the central government to implement the recent developed technology. Then as far as the extension activities are considered, our university is always emphasizing to conduct the different extension activities under the faculty of fisheries, like skill development program for the freshwater, brackish-water aquaculture, then village adoption, then farmer scientist forum, then lab to land program, farmers rally, field level demonstrations, then exhibitions, demonstrations, then trainers training program, and awareness of this recent developed technology to the farming community and fishermen community through the mass media like All India Radio, television programs, and so many other mass media. So like this, our university has did the very well work for the faculty, and they have given special attention for the development of the faculty of fisheries. Also, our students who are present today, they are present. Out of that, Dr. Deepak Chaudhary, who is the student of the first batch, and that Dr. Deepak Chaudhary, he is the director and proprietor of the Nishiendo Foods, which is at Verawali in Gujarat. So he is the first batch student who is the entrepreneur, big entrepreneur from our faculty. Then another one is Prabhakar Ghadge. He is working at Goa and his company is Maritime Seafood. Then another students, that is Rupesh Sarkare. He is an entrepreneur working specifically on ornamental fisheries, and he is having the Uttekar fisheries farm. Then Mr. Shashikant Khoth, who is the third batch student, and he is my classmate also, and he is having the business of private seafood industry, and he is doing the export business. Then Abhiti Jadhav, he is also doing the consultancy, having different entrepreneurship. Then Makarand Deshpande, he is also doing the agro processing industry. So many students from our faculty, they are doing very well in different fields, and specifically in the entrepreneurship development program. So I once again thank you to all of you. Once our Honorable Dr. Sanjay Sawant sir, Honorable Vice Chancellor, our Director, and our beloved DDG, Dr. Jena sir, for given the time, and you have taken the time from your busy schedule to give a guide to our audience, to our students, our teachers, and all faculty members. Thank you one and all. Thank you very much.
Thank you, Dr. Shingari sir, for your preface for this particular webinar on entrepreneurship development for sustainable fisheries. As you know, and sir has described in his speech, the journey of last 38 years is full of achievements and full of creating very good citizens for this particular country. There are many entrepreneurs, and major objective of this particular institute is to provide or to create the job providers. And therefore, we have chosen the theme of entrepreneurship development, and also this particular entrepreneurship development with the objective of sustainable fisheries. With this remark, we'll go to the next one. I request our Director of Instruction, Dr. Satish Narkade sir, to kindly address to the gathering. Dr. Narkade, sir.
Yes. First of all, I will like to say the congratulations to the College of Fisheries for entering into their 39th of the Foundation Day program today. And I also welcome Honorable DDG Fisheries, Jena sir, Honorable Vice Chancellor, Directors of the Dr. BSKKV Dapoli, Director Extension, Director Research, Associate Dean of the college, and the guest speakers, those who have spared their valuable time today to be with us. So on this occasion of the Foundation Day, for the DDG sir, I am submitting that this is the second Foundation Day in this month of August. The one College of Forestry's Foundation Day was there on 1st of August. And the theme for that Foundation Day was the forestry through the eyes of foresters. So those who have passed the IFS or the other exams in the forestry, or those who have excelled in the field of forestry, they were the guest speakers on that day. And today, the Fisheries College Foundation Day is there. And today, again, the theme is same. That is the fisheries through the eyes of the entrepreneurs or the ex-students of this college, those who have excelled in their field. So we have started this series this year, that looking at the particular discipline through the eyes of the experts or through the eyes of the students or the alumni of this college, those who have excelled in their field, whether it is in country or outside the country. But the fisheries faculty have chosen the three speakers, those who are settled outside, and they are the alumni of the student. So we are proud to say that our alumni are doing better and our college is also doing better. That can be proved with the help of these three alumni who will be speaking their experiences and about their knowledge and what they have earned from the college and what they have that has helped them for the future. And that can be a guiding lines for the students of this college. Though this situation, we cannot reach up to many, but I find that many students, they have joined today and they will listen to the experiences of these alumni and the entrepreneurs. Our main motive, as we know in the 5th INS committee, our major focus is on the development of skill in the students. So how they have earned the skills, these three speakers, they can share their experiences, that how they have earned the skills and how they have excelled in their field. So I wish again the best wishes for the College of Fisheries on their Foundation Day. And again, I am thankful to the guest of honor, Jena Saab, Vice Chancellor sir, and the guest speakers for sparing their valuable time. Thank you very much.
Thank you, our Director of Instruction, Dr. Narkade, sir. This particular institute has produced graduates and postgraduates not only from Maharashtra, but from all the states of Maharashtra, right from Kashmir up to the Kanyakumari, that is up to Tamil Nadu and Kerala. And even our three speakers, all the three speakers, one is from Andhra Pradesh, Dr. Nagamurli. Second, Dr. Baneshwar Singh, he is from Bihar, and Dr. More is from Maharashtra. So we have selected or rather we got the speakers from all the states representing their respective state and our nation. So thank you very much, sir, for your address to the gathering. And this is really a great opportunity for me also to welcome our Director of Research, Dr. Parag Hardankar, sir. If sir is there, because there was a continuous meeting from morning, and because of the continuous morning meetings, I think sir is not available. So I will request our Director of Extension, Dr. Bhave sir, to kindly address on the 39th Foundation Day of College of Fisheries Ratnagiri and the Foundation Day lecture series inaugural function of this particular college. Dr. Bhave sir.
Honorable Dr. Jina sir, Honorable Vice Chancellor Dr. Sawant sir, Dr. Shingare, Dr. Narkhede, Director Instructions, Dr. Shingare, Dr. Baneshwar, Sri Naga Murli, Dr. Amit. Chaudhary, could you hear me? Dr. Chaudhary?
Yes, sir. Yes, sir. Yes, sir.
Sir, all delegates and all staff members of the College of Fisheries and students. It's really a happy moment that College of Fisheries celebrating its 38th Foundation Day. So I congratulate them. 38th Foundation Day, it doesn't mean that it is the youngest one. Almost 36 batches passed out. Though it is youngest, it is from old culture area. When we say the fisheries, so in Darwin's theory maybe, or in any case in our Dashavatara also you can make out. Dashavatara also you can make out that first avatar was Matsya Avatara. And the land where this college is there is the sixth avatara, that is land of Parshurama, god of Parshurama. So here, so it is from old culture, the college where it is old culture and youngest college. College is known by its alumni, it is said. Generally people they said, Atma Jayate Putraha. How your alumni, that depends how that father is. So it's really a proud moment for you that the students from College of Fisheries are doing well, not only in India, but outside too. It's a really proud moment. And therefore, I proudly say that this College of Fisheries, as far as education is done very good job. Research contribution, it's really good. They have done very good work. It may be in cage culture. They have done good work in inland fishery, maybe basa fish, and so many. But when I took over the charge of the director extension 7 years back, I really come across the hot work these fisheries people they have done. And they have a capacity to give, rather they have a capacity to develop entrepreneurship in a good manner in Konkan, particularly in COVID era or past COVID era, as migration was the major problem in Konkan. But because of the COVID, reverse migration is started. From Mumbai and Pune, people are now coming back to their native places. And the question does arise before them and even before everybody, what kind of work we can give to them. But fisheries people, they are coming up well before us. They have given so many good technologies. It may be crab fattening, it may be prawns development. For that matter, Mulde were doing very job on ornamental fisheries. So that will definitely help us to give some work for the hands of the people, those who are coming from Mumbai, Pune. In future also, they will definitely do well. As far as the extension work is concerned, I am very much satisfied with the hot work this College of Fisheries are doing. Once again, I congratulate all the staff and students of the College of Fisheries. Thank you very much for giving me chance to put up my thoughts before you. Thank you very much.
Thank you, sir. And thank you very much for your address. Sir, you have mentioned about the reverse migration, and under your direction and under Honorable Vice Chancellor Sir's direction, already we have started working on, we have conducted near about last 2 to 3 months, we have conducted 3 to 4 webinars, and all those webinars were joined by all these people, those who are migrated from Mumbai. They have very good interest in different farming system, including the crab culture, shrimp culture, biofloc culture, RAS culture, all these technologies, all these people, they are interested. And I hope this particular faculty will provide the essential training which is required for creating the entrepreneurship among these migrated people from Mumbai, Pune, or other places. Thank you very much, sir. For this particular webinar, we have our beloved Deputy Director General Fisheries Science, Dr. J.K. Jaina, sir. We contacted sir, and sir has very happily accepted our invitation to join this particular webinar. Thank you, sir, for accepting our invitation in the first instance only. Thank you well, sir. I request you to kindly address to the gathering. Dr. J.K. Jaina, sir.
Thank you very much, Chaudhary Sahab, Dr. Chaudhary Sahab. Am I audible? Am I audible?
Yes, sir.
Okay.
Yes, sir. Yes, sir. Yes, sir.
Most Honorable Vice Chancellor of the Konkan Krishi Vidyapeeth, Dr. Sanjay Sawant ji, Dr. Prakash Shingareji, Dean College of Fisheries, Ratnagiri, esteemed speakers, Dr. Baneshwar Singh ji, Dr. Naga Murli ji, Dr. Amit More ji. I could find or hear also from Director of Instruction, Dr. Narkade ji, Director of Research, Dr. Haldankar ji, Dr. Babhe ji, and all the invitees, faculties, students, alumni, farmers, and entrepreneurs, friends, ladies and gentlemen. It is indeed a great honor and privilege for me to be here this afternoon. And first of all, I would like to thank Honorable Vice Chancellor Dr. Sawant ji and Dean of the College Dr. Singare ji for giving me the opportunity and inviting to this occasion. First of all, also I would like to convey sincere greetings to each one of you. Congratulations to each one of you, dear students, faculties, and alumni in particular, including the Honorable Vice Chancellor and Directors of Extension, Research, Instruction, and all for celebration of this 39th Foundation Day ceremony. And along with that, again, an opportunity to be here for this webinar, an important area on entrepreneurship, which you have been conducting. Last 4 months, yes, it has been a difficult times for all of us, but we have learnt a lot also. We were rather, I would say, not that much of conversant with this e-technologies, e-platforms. Not that e-platforms were not there all the time, it was there, we used to hear, never used to maybe using this, used to be using this. But last 4 months, it has become a new normal. And every day, at least 3 to 4 meetings we have been conducting under this VC platform. I am sure all of you are involved in that. Friends, it is a time for us to introspect also and have a SWOT analysis on everything, whatever we do. When I say that we need to analyze the last 4 months, the sector which has undergone in last 4 months, we have seen. I don't intend to discuss about that because this is a difficult phase, definitely. We have learnt a lot, and we will see that in future also if similar such situation comes, probably we are better prepared than probably earlier days. We will be hearing the 3 speakers who are your alumni, and it is a good opportunity for all of you. I am sure all of you need to be congratulated for that you got the opportunity to hear from your not only the experts, but they have been your alumni. Friends, it is just about I think last year in January, if I remember correctly, I had visited to your college. And if you see this time, again interacting with you under this VC platform after 1.5 years, so much of difference. That was also a seminar. This is also a seminar, Foundation Day, but totally different. There we could have a handshake with each other. We had opportunity to meet many people, and together we used to eat also for 2 days, and several things we learning lessons also we had. And today, if we see, probably you say that that opportunity is not there. But let us also take that what is that advantage of this platform over the physical meeting. If it would have been a physical meeting this year, also a foundation day like last year, probably you would not have got three of your speakers who are from US, both of them for Dr. Singh and Dr. More from US, and Dr. Murli from Ghana. I could understand. Definitely it would have been difficult. And probably it would have been also difficult for me to be there. And last year also Honorable Vice Chancellor Sahib could not come because of suddenly some meeting came. So you see the advantage. At a time we could able to see 60 people at a time. Maybe many more are there in other platform. I do not know. It may not be 60, maybe much more people are hearing to this discussion process. Coming to the two aspects, I do understand. I just spoke to Singare Saab. I asked whether I need to give a presentation. He told not necessary. And then I asked how much time you can speak. I can speak. He told maybe half an hour is better. So I will try to be completing my speech in within maybe another 25 minutes. But if I take more, please stop me. In between also, if you like that I should conclude, I would be not disappointed to stop definitely. Friends, when you talk on the subject of entrepreneurship, how important it is, all of us know, when we talk of the sector like fisheries, a new sector compared to many other agricultural sector and all. What we say every time, people say it is a sunrise sector. Yes, when you talk today about the fish as a food, we have been talking of fish as for protein security, fish for nutritional security, and today definitely we don't talk of fish for protein, we talk fish as a health food. So important. So it is very much necessary for the message to be taken to the people, that we don't talk fish as only protein or nutrition, fish as a health food. When you go to a doctor, he or she prescribes something, you need to be taking daily, maybe once, twice, thrice, so and so forth. So I say, if you take fish, if we can take this message, fish to be taken daily basis, probably there will be no health concern in coming days. And coming to the fisheries, we talked of fisheries means including fisheries and aquaculture, both capture fisheries and aquaculture together as a profession. We know the country like India, still we talk of fisheries or aquaculture for livelihood. We have been talking for the livelihood. But when you talk of fisheries today, it is totally different from that of 3 decades back or 4 decades back, what fisheries were. Today we talk every subsector of fisheries or aquaculture we talk of. There is a scope of entrepreneurship involved everywhere. When you talk of, for example, we take the pride to say that, okay, the sector is growing 6 to 7% per year, maybe last 30 to 40 years, probably unparalleled. Many, if you talk to other sector compared with the other agriculture sector, maybe very few sector will be there giving a consistent growth rate of 6 to 7%. And the last 2 years, again, the growth rate has been still higher, about 10% or so, the growth rate has been. We do talk of export increase and all. We have been successful to good extent, definitely. 47,000 crores of export is not a small joke. Again, India, in the target set in the next 5 years, the government of India has, as you know, that set the target of about 1 lakh crores export besides meeting the domestic demand. Probably the sector is looking for much more entrepreneurship in coming days. When you talk of again entrepreneurship of different subsector, as I told, this if you see the recent years, the entrepreneurship have been mostly on the seed production sector, hatchery front. If you talk of maybe 2,500 to 3,000 hatchery only in the case of freshwater carps, several of diversification is happening. Today we are talking of several brachywater and marine, both finfish and shellfish. When you talk of again seed production, we have more than 550 hatcheries in the brachywater sector. These shrimp hatcheries, all of you are aware about this fact and figure. Today we are talking of again several of the finfish hatcheries which we need to have. With the cage culture growing all around, all of you know that today, about 10 years back, we had nothing, whether East Coast or East Coast or West Coast. We did not have a single cage. Today, we are talking more than 3,000 cages at least installed all around. We have been talking of not only diversified species, but also very high value diversified species like cobia, pompano, silver, and grouper or snapper. All those we have been able to breed most of these important species today. So we are looking for more entrepreneurship in the coming days, mainly in the seed sector. I have been talking with several entrepreneurs over the days. People want to invest, but probably they don't have, at this point of time, they do not have confidence because they are not able to see a single hatchery in the private sector. If somebody want to start a cobia hatchery or pompano hatchery, they need to, they know that only in the government sector, 2-3 hatcheries, whatever available with CMFRI or RGCA. There is not a single private sector there. So they are probably a bit reluctant in that context. So it is very much necessary for us to connect with them, to have a confidence building with them. Probably as some of you were, Dr. Shingare Saab was talking, that some of the alumni, if they are coming forward because they are already educated or skilled, they are skilled already, probably if we can handhold with them, if some of them are ready to take up. I don't say that they need to be there, but I'm sure some of them definitely will be interested. So considering the fact that the country is looking in a big way for the cage culture in coming days, that is what next 5 years, all of you know, the target set by the government of India is 22 million ton from the present 13.4 million ton. It is a huge, about 8 million ton we have to add, if at all, whether it is within 5 years or 6 years or 10 years. But target set is 22 million ton, which is quite possible. This increase of 8 million ton has to come from the aquaculture. Mostly, all of you know the fact and figure, what is happening in the open water fisheries, whether it is inland or marine open water fisheries. So everybody is looking at the sector like aquaculture, whether freshwater, brackish-water or marine cage culture sector. So every sector has to take the responsibility to add something more than whatever we are producing. Yes, we have been talking of several diversified spaces. We have been successful in good extent. Today, if you just see the list of the diversified spaces in which we have been successfully able to breed or seed production in a commercial scale, we have been able to achieve more than 60 species. It is a huge number. And most of them, at least 30 to 40 species have been farmed in some of the location. We cannot compare with the neighboring state like Bangladesh or India, same, because Bangladesh is a so small country, similar to one of our state or smaller than one of the states. So any technology we are talking of today cannot be applicable for the whole country, that we are very much aware. Even one species or two species are targeted for even few districts, that is good enough also. And we have consumer preference, different consumer preference are so different. For example, West Bengal or Assam or any of the northeastern state you go. For example, this species like Pabda, Ampok Pabda. or binocular tests, even anavus, or even small, maybe minor calves like gonias. Nothing is sold today less than 500 to 600 rupees. Whereas this species does not have market in if you go to south or other places. For example, the morels, which are sold at 500, 600 rupees in the some of the states like Karnataka, Andhra Pradesh, or Tamil Nadu, may not have good market in other places. So depending on the consumer preference, when we talk of entrepreneurship, probably the proper guidance is so important. Yes, today with the transport facility, people say even if Andhra Pradesh fish can go from Andhra Pradesh to Guwahati or beyond, why fish cannot be transported from Gujarat to other places? Yes, it can be done, but why should we spend 20 rupees more during transport? Why not we target those places in the location where these are important or these are more preferred? So probably we need to, because today date, several entrepreneurs come to us, each one of us. Many may be coming to you in the college, being an important college of having a reputation of 38 years. Many entrepreneurs may be coming. So a proper direction to the entrepreneurs are so important today. Because if you see today, every day, at least every day, I get at least a 2 to 3 mail or WhatsApp message from some people. They want to start something. For example, the last week, Man Ki Baat, you must have heard, Honorable Prime Minister spoke about freshwater culture. Just after that, I started receiving several mails on that, because people, those who did not know, they want to start something. Similarly, recent years we have been talking of RAS system, we have been talking of biofloc system, as because many of the things are available in the what you call YouTube. Many of the people, they are seeing some YouTube and they are trying to invest. I know a couple of farmers or entrepreneurs who have seen from the YouTube, they have invested a lot, but because of they did not have background, because they did not get proper technical support, probably they failed to get whatever they wanted to, whatever was projected to them. So it is very much necessary for us, people like us, to not only to connect them, give them proper guidance, so important, because today when you talk of entrepreneurship, many times most of the people who are interested to invest, I found them, most of them are either engineers, doctors, or MBA person, or so and so forth. They have confidence with them, but they are not aware much of the technology. So probably a proper guidance are so important for them. Today we are talking of, for example, when you talk of entrepreneurship, as I was talking, the hatcheries, for example, shrimp hatcheries. We have about 550 hatcheries. These 550 hatcheries are producing today about 60 billion of maybe post larvae, which is good enough, to at present whatever is necessary. But if we are targeting to increase from present production from maybe 8 lakh tons of shrimp today to about 14 lakh ton of shrimp by 2025, just 5 years. Beyond 25 we are so much. Just 5 years, the target government of India has set a target production of 14 lakh tons, 1.4 million ton of shrimp. That means you need about 120 billion of post larvae. That means you need to have another additional, another 250 to about 400 hatcheries, new hatcheries. So if you consider maybe only shrimp, beside that maybe other fin fishes and so forth. So you think how much investment you may need for those hatcheries itself. For example, the carp itself. For example, today we are talking of, if we want to add another 8 million ton, that means mostly another 6 million ton definitely need to come from the carp culture sector. to add from the present production from 8 million ton of carp, for example, today we are producing to about another 6 million ton, how much more hatcheries are necessary? And today, for hatchery establishment, probably you don't need to give more technical guidance to anyone. With the technology available everywhere, every village front also, little hand holding is good enough. Little DPR, a small DPR availability with us would help the people. For example, if somebody want to say that, okay, I want to start a hatchery of 50 million capacity or 100 million capacity or 200 million capacity, how much I need to invest? What would be my recurring expenditure per year? And how many years it will take to get back my investment? A small, even the small 2 to 3 days discussion would help a lot. So probably I would say when you talk of in the future aquaculture only, seed is most vital. Today we know the problem of seed. It is not the quantity. Quantity we are getting it. It is the quality which are more important today. We know the issues still today. People are stocking fry in spite of the fact that we advocate fingerlings or juveniles. We have been talking about diversification. Still, the seed of diversity species are maybe available only in few packets. It need to be transported. This year in the COVID situation, you must have seen. All of you know that we produce about 200 billion of spawn, carp spawn. And out of 200 billion, about 120 billion are produced only by the West Bengal. Only by the West Bengal, about 60%. And West Bengal produce most of the spawn during about 60 to 70% they spawn they produce only during March, April, May, which was affected a lot. Not that they could produce, they could not produce anything. Almost the production was zero in West Bengal because there was no transport facility available. Demand in other places was not there. So we foresee about 30% less demand this year, overall demand. So in the coming days, probably we need to be thinking not that only from seed will come from West Bengal, then only we will start. Probably we need to have a more discussion with the entrepreneurs in different regions and suggest them what species they should start. Because many times, sometimes also some people comes with a very big idea. Sometimes some people say that I want to start 500 hectares at a time. We need to suggest them properly. They need 500 hectares at a time or 5 hectares to start with? Maybe they can have a plan for 500 hectares, but let them start with only 5 hectares. First year, get confidence, then get convinced, then probably expand. 1 year is not a big deal, definitely. So probably it is very important for us to give a proper advice to the people. Today, when you talk of entrepreneurship, we know that there are several areas we can think of entrepreneurship. For example, feed. Today, if you see the feed sector, whether it is sinking feed for the shrimps or even the catfish feed or carp feed, floating carp feed, whatever, most of these feeds are produced mainly from the Andhra Pradesh recently and Chhattisgarh, and being transported to all the places. So does it mean that other places it is not there? It is not important, or because it is available plenty, it is easy for us. Probably we are transporting. Coming to the again the demand, increasing demand of the feed in the coming days. If we are looking for increasing the production, maybe another 6 million ton or so, definitely there will be demand for feed. So the entrepreneurship, when you talk of entrepreneurship in Andhra Pradesh or Chhattisgarh, most of the entrepreneurship in the feed industry are large scale industry. Probably it is necessary in many places we need to talk to them for medium term, small scale feed plants and so forth. there are few areas of entrepreneurship which are growing at present. Probably one is ornamental fish farming. If you see the recent years, there has been so much of increase in demand of the ornamental sector, which is growing at 14 to 15% per year. 14 to, of course, we are when you talk of ornamental fish market, we are very small compared to maybe whether it is Singapore or even our neighboring country like Sri Lanka. But with the present growth rate happening, we foresee several more entrepreneurship coming into the sector, because this is a technology which we call scale neutral technology. If somebody does not have more money, 50,000 rupees you want to invest, he can start with the backyard unit to start with. If somebody have big money, they can start with high value species or high value marine species like damsel, clownfish. Today, a clownfish are sold at $4 to $5 in the international market. With the not that technology is not available. Today, we have the technology available for more than 20 species of clown and damsels. And not only clown and damsel, some of the designer clowns also, new strains or new varieties of clown. So we have, I would say that sky is the limit at this point of time. We have been talking of several of the brachyceratous species, for example, even some of the species, for example, the species like Scatophagus or even the Atropus, which we consider not only as a food fish, but also a ornamental species. So we have a huge scope on those, some of the areas. Some of the other area, what I find is probably entrepreneurship would happen. One is health sector. Probably we had not given that much importance in the previous years. For example, diagnostics. When you talk of fish, probably nobody knows whether we should be using diagnostic at all. When we say diagnostics, everybody think about the FMD or even PPR or goat pox, sheep pox, blue tongue. All those people think. People think in the fish we don't need to have a diagnostic. People think that we cannot have vaccines for the fish. But if you see for example, most of you must be knowing for example, cage farming in Norway. Each and individual fishes are vaccinated. You must have seen. So probably days are not far. We need to be little forward looking. With the new technology coming in, with the high value species we are talking of, for example, marine cage culture, there is a possibility to use those. We need to be prepared enough for many of these things. For example, several of the formulations, in spite of the fact that, for example, the EUS, epizootic ulcerative syndrome, last 30 years we have been talking of. Some formulation is there here and there. Probably we have not given that much focus over these years, but after this COVID situation, everybody today are aware how health is important, whether it is human, animal or fish or any other. Health is so important. And the loss, probably people are not aware because we have not done loss estimate. We do not know how much is lost in when you talk of loss estimate, people only say how much amount of fish died, but how much indirect loss because of the growth retardation. We have never tried to analyze those. So it is time for us that we have also assessment to study on the health aspect, every aspects of the health. We have a national disease surveillance program in place, and your college is also a partner of this surveillance program. We want to see that this partnership grows in the coming days, definitely. I think I have taken about 25 minutes. I must try to close in another 2-3 minutes, definitely. I won't intend to take more time than the allotted time, definitely. But as I told, this sky is the limit for when you talk of fisheries. Just talk about the shrimp farming. We are talking that 1.2 million hectare of potential area. How much we have been using? Only 1.7 lakh hectare. That means only about 18% of the land area we have been using. That means we have a huge scope. Again, the places like Punjab, Haryana, Rajasthan, where you have a saline affected areas. We have been successful to good extent. Today, last 5 years, we have been able to at least have adoption of about 1,000 acres of land for shrimp farming. And let me tell you, average net profit what farmers are getting about 3 to 5 lakh rupees per acre. I don't think any other farming practice can give such net profit. But we know that the risk involvement, all those things we know. Without risk, there cannot be more profit. If somebody want to have more profit, they need to take risk. But risk can be minimized with a better preparedness, better technical support. The future days farming, when you talk of future farming, whether it is agriculture, animal livestock, or fisheries, our farming over the years were targeted only production and productivity. That was the major target, and we have been able to achieve. Today, this production on productivity, what we have been able to achieve, it is because of mainly input-based farming. Mainly input. But future farming has to be not input-based farming. Future farming has to be knowledge-based farming, which all of us know from where the knowledge will come. Knowledge has to go from the people like us, the institute, colleges, and so and so. And how people will be utilizing the knowledge, those are important. Today, the objective before us are manifold again. Maybe when we got independence, maybe about 30 years back or 40 years back, we had only target of production and productivity increase. Today, our target is besides production and productivity, that is there, but quality has become so important. All of you must be knowing last year, because of the formalin usage, how much hue and cry everywhere. And immediately we came out with the diagnostic kit for formalin detection. Just a paper-based strip of 1 rupee or 2 rupees, we could able to know the adulteration with whether ammonia or even the formalin. That is what the entrepreneurship can come on all those small sector. Even today we are talking of, we are trying to review a few days back, what is that waste to wealth. I was trying to analyze waste to wealth. Every part of the fish we have been, whether it is chitin, chitosan, or any other things, how best to use. So as I told, we have several systems, several enterprise in place. We have knowledge with us. We have scope is unlimited. As I told, today not only production, productivity, quality, safety, livelihood, farmers' income, maybe environment, doubling farmers' income, everything is important. This is in aquaculture, but in the marine front also similar, with the fisheries production coming down in the coastal areas at less than 50-meter depth. We are talking of deep sea fisheries, for example, today we are talking of yellowfin tuna. The amount of tuna we produce about 40,000 tons, for example, yellowfin tuna, that are sold at only $1.50 per kilo. Because the quality of at the landing place, all of you know, because Ratnagiri is a beautiful landing place, I could see, wonderful place. But you know, the international market of yellowfin tuna is about $5 to $7 rupees, $5 to $7 per kilo. So there has to be several intervention, entrepreneurship need to come on several of these areas. We have been discussing several of the diversification we have been talking of today, whether ornamental, seaweed, etc. Our Honorable Prime Minister all the time have been speaking of diversification with seaweed, ornamental fish, and several other areas. So friends, I don't intend to take more time, as I have completed my allotted time. Thank you very much for your patience hearing. But I would love to answer anything. If you have any response to have from me also, I will be available for at least another half an hour. Till definitely Sawant Saheb is there, I will be there. Because I don't commit that I will be there till end, but I will try to be there as much as time as possible. It is wonderful speaking to all of you. As subject is so vast, we can go on speaking over hours together, but that is not right also. Thank you very much for the opportunities, Dr. Sawant Sahab, for the invitation, and Dr. Singari Sahab, all the faculty members, students. Congratulations again, good wishes on behalf of me and on behalf of the Fisheries Science Division of ICR and on behalf of ICR on the occasion of 39th Foundation Day ceremony, and wish you all the best. Stay fit, stay healthy, because we have to, again, this situation may remain for some more time. We need to be there in safe. Thank you very much. Thank you so much.
Thank you, sir. Thank you, sir, for your very wonderful address and encouraging all our students and postgraduates, graduates and postgraduates for starting the entrepreneurship in the fisheries sector. Sir, as you have said, the journey of the fish has started with the fish for the protein-rich fish for the poor. And now the journey has reached up to the fish for the healthy food. So that is true, sir. And the growth rate of our fisheries sector has been increasing. It is constant since last last so many years, but it is at 6 to 7%. Last year it was at the 10%. You have emphasized the need for establishing more and more hatcheries so that farmers, they will get seed. From our faculty also, we have always have the approach to provide the seed for the farmers. Many times the inquiry comes for providing the seed and feed. Thank you, sir. Also, you have said about the diversification in the species, and this diversification in the species is being dealt by our faculty by having the research in different aspects of biological aspects and then the hatchery aspects. Hand-holding to the farmers and the fishers is very important. You have emphasized the need for having the hand-holding so that the dreams our Prime Minister has said, the Prime Minister Matsya Sampada Yojana, and through this Matsya Sampada Yojana, whatever investment of rupees 20,000 crore rupees is going to happen in coming 5 years, this particular investment will be a real boost for the fisheries sector to achieve all its targets. And all our students, graduates and postgraduates, all our efforts are to make them competent so that they will have the entrepreneurship in the fisheries sector. So thank you very much, sir, for addressing and motivating our students, our faculty members on the occasion of 39th Foundation Day of this particular college. Thank you, sir. After this, sir, I request our Honorable Vice Chancellor, Dr. Sanjay Sawant, sir, to deliver the presidential address for the today's inaugural function of this particular webinar, a webinar on entrepreneurship development for sustainable fisheries. Sir, already our guest lecturer has delivered the lecture, and Dr. Baneshwar Singh, he has already delivered the lecture on the bugs and bacteria to the rescue of forensic and the fisheries science. The two lectures are remaining, and after the address of Honorable Vice Chancellor, we'll continue with respect to both these lectures. So I request our Honorable Vice Chancellor, sir, to kindly address on this particular occasion. Thank you, sir.
Thank you, Chaudhary Sahab. Honorable DDG Jina Sahab, welcome to KKV. KKV's jurisdiction, and it was very, very interesting and a pleasure to listen to you. In fact, I congratulate our organizers. They have brought one of the most appropriate guest of honor, and you gave them only small time. He could have spoken more time than what he actually. I was listening to all current thoughts about fisheries and entrepreneurship in general in fisheries. Thank you very much, Dr. Saab, for joining our webinar and this program. We have 3 speakers on the line. Dr. Baneshwar Singh, Shri Naga Murli, probably Managing Director of Volta Breams Limited, Ghana. And the third one is Dr. Amit More, Assistant Professor from Auburn University, Alabama, USA. So all the three I welcome, and thank you very much for accepting our invitations for this webinar. My university directors, Dr. Hardankar, Dr. Bhave and Dr. Narkhede were online, and they also spoke at this occasion. I first of all congratulate the team of Dr. Shingare, along with his all staff, heads of the divisions, and even some of the students and alumni who are there attending this program for celebrating 39th foundation day of their college. And it must be a proud moment for all of you, because most of people whom you have invited are your alumni, particularly the speakers in this webinar. And Dr. Shingare also gave a fairly good account of their achievements over last few years, historical background and other things. It was really a proud moment in that background also, that you have more than 1,000 graduates, few hundred postgraduates in the field of fisheries, and they are spread across the, they are not only from Maharashtra, they are spread across the different states in country, and they are working outside the country as well for their post-graduations or any other assignments. And it should be a great moment for your college. And I am particularly very happy immediately after my joining as a Vice Chancellor, probably this university underwent accreditation program, ICRs. And there were grading of the colleges, and I was very happy to see that Fishery College got A grade. And they have scored good among all other colleges of the university. It is also a good thing. I request all your staff members and team, Dr. Shingare, that you maintain it for coming future also. Coming back to your webinar, you are mainly discussing about entrepreneurship, particularly for a sustainable fisheries in the region. It's a good topic. Actually, Dr. Narkhede made a mention that they have got a series, all the college foundations. We are talking about subjects related to entrepreneurship. Lot of good things were narrated and highlighted by our Honorable DDG regarding what are the possible entrepreneurships. One thing which he probably mentioned about seed production or hatcheries, and he definitely gave you a good amount of target that this year, coming future, we have huge scope for increasing fisheries production in our country, and which will require lots of seeds. In this portion, the one point what you mentioned is that most of the seed is coming from West Bengal. And the situations like lockdowns of COVID, we had a lot of difficulties, and we are continuing to have a lot of difficulties in procuring fish seed from outside state. And we should look into it, and we should encourage lots of entrepreneurs to develop various kind of hatcheries within our state as well as maybe in the region, Konkan region. Because we definitely have lots of scarcity of fish seeds in our area. One thing I would make a mention here, that university on their farm itself is trying to develop inland fisheries in a big way. We want to demonstrate for the farmers. We have more than, I believe, about 16 or 17 water tanks in the campus itself. Maybe on a Dapoli campus or a agronomy department, and in Wakoli farm. All put together, we have about 16 tanks, water tanks for irrigation, basically. As on today, to my knowledge, only 2 or 3 are being used for fish culture. Rest all only we gather water and use it for irrigation. So we have a thinking this year that by say 2021, we are entering into 58 years of existence of our university. So we have made a resolution even actually today only, that most of our water tanks should be used for aquaculture, for inland fisheries. I do not know much about fisheries as such, but what I have been hearing most of the time, people are going only for tilapia. I don't know what is that tilapia. Tilapia, I only know that it grows rapidly. It multiplies rapidly. If you allow it to multiply rapidly, it can. It doesn't develop size because of the high population. This particular fish, many entrepreneurs want to grow, but many of the occasions I have heard that tilapia is dangerous. It is sometimes referred as aquatic weed, rather, because it multiplies faster. If you allow this fish culture to go into our natural water resources, it may not allow it. It may grow much rapidly than what it should, and may suppress our local fish species. Like many, we talk about rohu, katla, and many carps and other things. So we should be very careful in promoting tilapia. This is what I seriously feel. Dr. Amit, Dr. Naga Murli will be talking about tilapia, various strains of tilapia into this webinar. We should be seriously thinking, it is not only earning more, you have to think about environment and future of the fish industry. So if it is really good that tilapia can be cultivated in many areas so easily, you have to take certain precautions that it will not go into natural water bodies like your rivers or maybe dam reservoirs and so on. So in that context, do we have sufficient hatcheries in case of our own regular species like rohu, katla? If they are not there, we should be encouraging some of the hatcheries in our area as much as possible. You may also mention of cage cultures. Now, cage cultures are coming in larger water reservoirs and even in sea water nearby area. I do not know much about it, but DDG made a mention about that we should be encouraging entrepreneurship by using cage cultures. If it is true, I don't really see much of cage cultures in our system. They should be there available in large quantity. while just closing your speech, DDG also mentioned about some of the waste utilization, where now we are talking about shrimp culture in a very large quantity. The chitosan is one of the major waste product, a good product you can make out of shrimps, I believe, shrimps waste. And this college at Ratnagiri has developed their own technology to isolate chitosan out of shrimp waste. It was running. Some of the small entrepreneurship was being developed around that region, but everything has stopped, saying that chitosan has no applications and there are no buyers. But I am seeing that agriculture has got plenty of application of chitosan. And I personally was using it for plant protection. And it is a recommended material for various purposes of plant protection in grapes, where I actually spend quite a bit of time for plant protection. I remember that now every day we are using sanitizers. The CIFT have developed a technology using chitosan. They have some hand sanitizers. Can we really think of, because this is something which is coming up, we have increased use of hand sanitizers. Could be we develop entrepreneurships again in by utilizing shrimps and its waste for production of chitosan. Recent times, our college has been concentrating on a box type of structures for fattening of sea crabs. And that technology, we are getting a feeling that it will give lot of earning by to the entrepreneurs who really practice it. In the process of fattening, the crabs molds and gives out its cover, its coat, and that can be a easily available waste material coming from that crab culture technology. Our university people are also trying to isolate chitosan from this crab waste. And we are trying to see what are the different applications of this particular crab chitosan, how that can be used in our system. If this complementary technologies are developed, because even in processing now, everywhere we talk about zero waste technologies are important. So in that case, this chitosan technology appears to be interesting for me. When we think really about the social factor, recently we had Nisarga cyclone, and it has destroyed many of the coastal area, including the assets of fisherwomen, fishing families. And those people, we should be thinking now that what are the entrepreneurship we will be developing for them for sustaining their families, particularly for small families, fishing families across the seashore. We have a long seashore, and this particular aspect we discuss somewhere. It will be of use. Another thing, due to COVID, if you remember, if you know, most of our people, what we call them Chakarmani, used to migrate to the nearby cities, particularly Bombay, Pune, and so on, for their earnings. See, their earning at their own in their own villages were not enough earlier, and that's the reason why they migrated. And they migrated there for their earnings, but COVID lockdown have destroyed their all dreams all of a sudden. Most of the people have become jobless. Some people even walked when they were not allowed to use any way of transport from Pune, Mumbai to their own villages. That itself indicates that their existence in city was so difficult and they were so helpless. But now they have come back to their villages, and we are looking what kind of an entrepreneurship we can provide them, so that after the COVID risk or COVID problem gets over, they will not go back to villages or from their villages to the cities for again their earnings. A few educated, maybe who are doing a better job, let them go to cities and earn money. But many of the people who are not having large amount of skill, or they are going for physical labor or similar kind of work for their earning in cities, we must be able to stop them back to our villages. So entrepreneurship development is should be targeted for particularly those people who have returned from cities back to their own villages. Many of them may be interested in entrepreneurship related to fisheries. You should target them. You should develop some programs, training programs, skill development programs, specifically for those who have returned from cities during COVID lockdowns. That will be very useful, and university, if able to do this, we will be doing very good service to our Konkan region. So let us see that all this kind of entrepreneurship development happens, and we will be able to say few similar entrepreneurs we have developed during this period. Actually, you all are waiting eagerly for listening to two next speakers. So I have already taken more time. Probably I might have got encouraged by a good talk given by our honorable guest, Dr. Jinnah. So thanks a lot for listening to me. Hope you would incorporate certain things which we have suggested during your forthcoming programs. I wish again all the best for your celebrations, and congratulations for completing 39 years, because as you are celebrating 39 Foundation Day. Thank you for giving me time for sharing my thoughts. Thank you very much.
Thank you, sir. Thank you, sir, for very excellent migration guidance from your side and presidential address to today's webinar on entrepreneurship development for sustainable fisheries. Sir, as you have mentioned about the climate change and cyclones in Konkan region recently, sir, today is also we are facing cyclone-like situation, and about 50% participants are less due to that. In Zoom, about 55 people they have joined, but on the YouTube, more than 100 people, they are still holding their position in the YouTube. Our expectation was about 200, but because of from all the sources, we are getting the information because of heavy rain and cyclone line situations, they don't have the internet connectivity and like that. Even today morning, our Baneshwar Singh, he has also informed me that there is a cyclone in Baneshwar's place. So whether he will be able to join it or not. But because of the God grace, everything is going fine. We are doing well with respect to our this particular seminar. Sir, in your presidential address, sir, you have talked about the waste utilization, and yes, our faculty is concentrating definitely on the utilization of shrimp and crab waste for the chitin and chitosan. Sir, you have mentioned about the crab fattening. That is a new program which our faculty has taken, and definitely in few years we'll get very encouraging results. Many entrepreneurs, they are coming. Many migrants, those who have already migrated from Mumbai during last 2 to 3 months, okay, they are attending our webinars, our skill development program. We'll specifically plan a skill development program for the migrants or the people, those who are affected due to the COVID, sir. Sir, you have also mentioned about, you have cautioned us, because one of the theme of our seminar is sustainable development of fisheries. Sir, when we are concentrating on the tilapia, so when tilapia, if it escapes to the natural water, definitely it is going to create havoc in the rivers, streams, especially 24/7 hours. Our streams are flowing in Konkan region, and all such streams will be highly affected if the crab is, tilapia is escaped from our culture system to the natural system. So we will be definitely including this particular point in our future programs, taking care about not to escape the tilapia in the natural resources. Because we want the development, but what we want is the sustainable development of marine fisheries. We want jobs, but jobs should be created by having the sustainability in the fisheries sector. That's the basic motive. With this particular motive, we are progressing. Last year also, we have organized the international seminar on considering the sustainable development in our agenda. Sir, thank you very much for sparing your time. I know, sir, since morning you are fully busy with the meetings, even you could not take your food up to 2:30, I think. Sir, because of that, we have postponed our inaugural function. But thank you very much, sir, for joining our program and giving a presidential address on the 39th Foundation Day lecture series of College of Fisheries, Ratnagiri. And not taking much time, I will be handing over this particular now next session to the Naga Murli. I'm getting so many messages from my friends, okay, that they are also very eager to listen to Amit More and Naga Murli, because both of them, they are doing very excellent work in their respective field. Both of them, they are entrepreneurs. They have started their own companies. And within a short period of a time, in last 15 years, their entrepreneurship has reached up to the international level. Okay, sir. So next, our speaker, this particular inaugural function, I think it is over. And I thank all the participants for this particular inaugural function of 39th Foundation Day of College of Fisheries, Ratnagiri. And technical sessions, sir, one lecture already we have completed from 2 o'clock to 3 o'clock. Dr. Baneshwar Singh, he has completed his lecture on very interesting topic, that is the bugs and bacteria of interest in forensic and fisheries sector. So that was very interesting lecture Dr. Baneshwar Singh has delivered. Now I request Mr. Naga Murli to, I think, to go for his Foundation Day, second Foundation Day lecture series, that is the trends and techniques in tilapia farming for successful entrepreneurship. Mr. Naga Murli is the Managing Director of Holtas Brems Limited, Ghana. Naga Murli, he was born in the family of aquaculture as a family occupation. From the age of 14, he had extensively involved in the fish and shrimp farming. To add more to the professionalism and the investment, he took up the fisheries science as a career and completed his master degree in the fisheries science with gold medal in 2003 from Dr. Balasaheb Sawant Krishi Vidyapeeth, Dapoli. He did his BFSc from Andhra Pradesh, and then he joined our college for MFSc. He did his MFSc in fish processing technology, but now he is in aquaculture. So the streams keep on changing.
Okay. Sound.
Construction design. Project planning, site survey, construction design, making the DPRs, construction supervision, equipment procurement, installation, hatchery setup, grow out in the ponds, cages, RAS, water quality and microbial analysis, post-harvest technology. All these things are handled by him for his client and also for his own company. So all the issues which are of concern today, now he is handling right from Ghana up to the India. He has a wide experience and exposure to the tiger shrimp, venomous shrimp, tilapia, Indian major carp, pangasius, African catfish species, hatchery, and grow out. Even he is having his own thoughts on the tilapia hatcheries. I think he will tell in his lecture, he can be contacted for any sort of a consultancy services in the field of aquaculture for any warm water species. That's the confidence Mr. Naga Murli is having after the experience of about 15 years of experience in the aquaculture sector in Ghana and also in India. So I proudly welcome our alumni and successful towering personality at the international level, Mr. Naga Murli, for his talk on the trends and techniques in tilapia farming for successful entrepreneurship. Thank you.
Yeah, Mr. Nagamurli, welcome. I'll take just a 1 minute. I may not get this opportunity later. I wish to invite Dr. Jina Saab to physically visit our university college within maybe a year or so. So we will be able to get the benefit of your vast experience in the field and guidance on further development of our college. So please do come. We will invite you later.
Sir.
Yes, sir. Yes, sir.
Sir,
Yes, sir.
Sawant, sir.
Sir,
I think we should invite him during 21-22, sir, for our
50th
Golden Jubilee year, sir.
Golden Jubilee year.
We'll invite him, sir.
So you are now formally बाद में करिए, but अभी yes कहिए.
Thank you, Sawant sir, for invitation. Let me tell you, last year, January, I visited the college. I got opportunity to not only attend the seminar and give my whatever presentation, but also I visited most of your facility, even I think as I remember, one ornamental unit, then also coast and all. But definitely I will love to visit again and again, not once, again and again, not only in the college, but also university. Thank you so much, Dr.
Thank you. I'm sorry, Mr. Naga. Please continue.
I will also be there for some time. Then I have to go for some other meeting.
Okay, sir.
Thank you so much. Thank you so much. Thank you so much.
Thank you, sir. Now I hand over to the Nagamurli, sir.
Good afternoon to the respected dignitaries and also to Ketan sir for organizing this webinar and giving me a chance and to be part of the webinar and also to present my topic. First of all, I would like to remind the days which I spent in Ratnagiri College, which was a game changer for me and it's a pathfinder for my life. I joined Ratnagiri College at the toughest part of my life. At that time, I lost my father in 2000. Then our shrimp farm was totally in losses. And it was not easy for me to join MFSC and continue MFSC. I joined and I was about to discontinue also. And at that point of time, the support given by the staff, especially I have to mention VP Joshi sir, VR Joshi sir, Shirdankar sir, Mohite sir, and all the staff, the support which they gave to me to complete my masters and to excel in my life, that was, I can never forget that. The confidence which they built me made me back again into the life, again back into entrepreneurship, a lot of confidence. And I also thank Ketan sir for his excellent notes on economics, which I still hold today and made as a template on Excel, where I use in each and every of my project to analyze the project and to assess the project. And I will also tell to all my juniors, the notes, the records, what our lecturers are giving, they are not just to read and pass out. They are the guides for our rest of our life. Like if it is engineering, or if it is processing, or if it is like economics, they are all the guides for our life. So don't neglect them. Keep the records and keep the books with you for the rest of the life also. Now, when I come to the topic of today's entrepreneurship development for sustainable fisheries, first of all, we all read the proverb that give a fish to a man, he will eat, teach about the fish, he will eat for his life. But how many of us do follow that? And secondly, there is another key proverb about our profession. Fishery starts where civilization ends. So before joining fisheries, if we understand this proverb, we will not have regrets and complaints about our profession. We have to work with the least educated people, least civilized people, and at the lowest infrastructure facility. And that is our profession. If we understand our profession, we have no regrets. So I would like to appeal to all my juniors and other people, like, know the truth that, and also like we should not stop. We should stop complaining about our jobs, and like, it's not a white collar job. And COVID-19 has taught us, not only us, to the whole world, many lessons and opened so many opportunities. And as I say, everything happens for good, and there is a cost, and COVID also has a cost. We cure nature and we go back to the self-sustainability and self-reliability, which we are now, the fishery sector is going into. And our great India under great leadership also giving an ample opportunity for entrepreneurship in the fishery sector. I wish that our alumni will make good use of these schemes and make our college and our nation proud to play a very good role in the Blue Revolution. Now, in the post-COVID situation, because of the reluctance of importation of seafood, especially like tilapia products from China, a huge opportunity is in front of us to occupy that sector, which is like a multi-billion-dollar trade is ahead of India, and only India can replace China in terms of export of tilapia fillet. And like I have been seeing the speakers talking about tilapia in various ways. Tilapia is the second largest commodity exported all over the world after salmon. It's a multi-billion-dollar fish. And India never entered there because of saying that we cannot compete with Chinese prices. But now the world is looking for an alternative, and it is our time. And to make up the export revenue, it's 1 crore, something like, $1 lakh or something like that. Only tilapia can do that magic, not any other species. Only tilapia can do that magic. And nowadays, in India, many aqua projects are encouraged for cage culture, RAS units. But why are we doing that? Only like, for justification, it is highly land cost. Land cost is high, and that's why we are doing cage culture. That's why we are doing RAS culture. And another thing, like if you are looking at the domestic production, we don't eat tilapia pangasius more in our country. We eat carps. When we are preferring carps in the domestic market, can we grow the carps in the cages? Can we grow the carps in the recirculated systems? We can't. So I think there should be some approach or some fine tuning of the approach of the promotion of this entrepreneurship according to the species demand market. Now I would like to start my presentation. So trends and techniques in tilapia farming for a successful entrepreneurship. You can see the logos of my company. The first one is like, Mansui Fisheries Private Limited, which I have registered in India. I have registered this company in 2019 and looking forward to do a little bit of more business in the coming years. And the second one is Volta Breams Limited, which is registered in Ghana. And this company is involved in tilapia hatchery, grow out, and black tiger shrimp farming. And the third logo is my value chain, app-based value chain, Hook Your Fish, and that is a logo for that. So I'm occupying the whole value chain, hatchery, production, and sales. Now, as described earlier, like my name is Nagamurli, Indian living in Ghana from past 12 years. First, I'm a proud fish farmer, then a fisheries professional who did masters from College of Fisheries, Ratnagiri, a gold medalist in my masters, a qualified assistant professor too. And we are known as Water Breams Limited in the Middle East and Africa, and as Mansfi Fisheries in India, which are started in 2019. And our main activities are tilapia hatchery, cage culture of tilapia, pond culture of tilapia, and tiger shrimp farming, app-based supply chain of seafood. And what is our specialty in our projects? We are known for the best aquaculture techniques and practices, and we're also known for using the indigenous resources for most of our project establishment work. So we always depend upon the local infrastructure, local materials, and we always try to encourage the industry on that basis. Our expertise of water breams in fisheries, expertise in fabrication of cages suitable for large water bodies, medium size of water bodies, expertise in setting up of hatcheries for Indian major carp, African catfish, pangasius, tilapia, and shrimp, expertise in post-harvest technology like fish processing, global trade, and established distribution channel, well-equipped engineering expertise to handle the construction process. Like in the future slides, you will see how fast and how economically we did the construction of a farm. A well-equipped team of experts in fisheries to handle the various production systems. And this is my profile. I have passed out my MFS in 2003, and later I worked in nurture aqua technology for 1 year. Then I worked for the Pfizer as the first aqua service officer. Then I worked in Alltech Biotechnology. Then I came to Zambia, Savannah Streams Limited, and later I joined Triton Group, which is a Mumbai-based group in West Africa. And in 2013, I started my career as an entrepreneur, starting Ulta Breams Limited in Ghana. I always analyze my business in 4 ways. First of all, what is the core strength and expertise? What is my strength? What do I know? Where should I invest? Then what is the strength and weakness of the business? Like I want to go to tilapia. What is the strength of this tilapia business and what is the weakness of the tilapia business? And what should we start with and what should be our final goal? Like my intention was to start in the low capital. I started with tilapia hatchery, and later I started growing in that sector, and now I reach a very big farm. So what strategy to follow and how to execute and what are the financial implications? Before starting out the project, knowing the financial implications is very, very important for any successful project. If you don't know the financial implications and how to execute and when to execute, definitely the project will get doomed. This is my first project, my own farm, which is in India, which is a 25-acre shrimp farm, which was constructed in 1991. And after completion of my MFSC, when I joined job, this was the farm which I managed in Nellore, which is a freshwater borehole-fed shrimp farm in Nellore. And this is the third farm which I managed in Nellore, which is a creek-fed black tiger shrimp farm. This is also for the same farmer, which is the earlier one. And this is my fourth project in Zambia, where I constructed a tilapia hatchery and ponds on the Kafue Flats of Kafue River. And this is my next project, which I did in Ghana, which is a 320 cages project, a tilapia project, with a hatchery at almost 120 meters at mean sea level, with a slope difference of 8 meters. And I have created a staircase ponds there on the top of a mountain without leveling the whole site. That was a unique construction which I did there in a very economical cost. And now this is the current project which I am doing, which is a JV between my company and other companies. And this is a 100-acre tilapia and black tiger shrimp farm. And we have a plan to expand to 1,000 acres here in Ghana in next 5 years. And this is the other project which I handle. This is a fish processing plant. And the fantastic part of this is this Elmina fish processing plant is funded by the Indian government Exim Bank. The whole fund has been given by the Exim Bank. And this is another circular cages project done for I did for a UK company. And other consultancy projects which I did was like construction of a shrimp farm in the Jordan Valley of Jordan. And I'm a consultant for the Oman government for a due diligence of a fish farm investment opportunity in Zanzibar. And I'm also a consultant for the Ivory Coast government for a program of transformation of aquaculture in Ivory Coast. Now let us go to the history of tilapia in India. Tilapia is a common name of the fish which belongs to the Oreochromis genus, and it is having a variety of species. Among all, Niloticus, which is most commonly known to all of us, which constitute 80% of the cultured tilapia. And other important species are Mozambicus, Andersoni, Mochimori, Macrochir, Rendali, Aureus, Zilla. There are so many species which we not whatever I mentioned. There are so many other species. In the beginning, what we all have been taught or know is that Oreochromis mozambicus, which is called Mozambique tilapia or black tilapia, came to India through China, which has spread all over India. And because of its enormous breeding capacity, fast propagation, black color, poor taste, Indians started treating it as a wheat fish. And even now I heard from the other speakers mentioning tilapia as a wheat fish. But tilapia is not a wheat fish. It has a multiple strains. We need to know which strain we need to select and how to go ahead with that strain. Even today, with much more advanced superior species of tilapia of Oreochromis niloticus, we have strains like Chitralada, Gift, Beginning, and we even have red tilapia also in niloticus. And still Indians look this as a wheat fish, which is a main drawback in developing the local market. And the fear of expressing if tilapia escapes into the natural waters, it will propagate it. But there should be no such fear, because all the commercial aquaculture of tilapia, we all do mono-sex culture. We all do only male tilapia culture. We sex reverse the tilapia fingerlings and we culture them. So even if this fish escapes into the wild, they cannot breed or they cannot develop into progeny. Only they will grow, and at certain stage they will die. So they will not pollute the waters and they will not become an endangered species for the other species. So strictly commercially, tilapia is done as a monosex culture, which is not a threat to the environment. And tilapia is called as aquatic chicken. Tilapia is called as wonder fish. And that's why it can create wonders in aquaculture. Apart from shrimp, tilapia has the potential to take the Indian aquaculture to the peak level in exports. An overview of Indian tilapia industry. The first tilapia introduced in India is like Mozambique tilapia, which I mentioned earlier in 1952. And the original introduction population of tilapia zille, then other species which all came. And in 2007-8, first time Niloticus has come to India. And the government of India is now supporting Niloticus and red hybrids also. And tilapia industry has taken over commercial picture in the year of 2006-2007 when Ars and Biotech has come up with a hatchery and production. And I have started a tilapia hatchery in India in 2006-07, and we have been looking for a good brood stock. And surprisingly, I found the best brood stock in the river Ganges at Allahabad area. We traveled all the way from Andhra to Allahabad to collect that tilapia. And then we brought it to our farm and we started breeding that. And we started inquiring, how did such a good tilapia gift strain has entered Ganges? Then they said some crocodile farm brought tilapia to produce tilapia as a food for crocodiles. It escaped into the river and then it has occupied. So the restriction, the restriction of the rules has to be made clear that tilapia should not be bred in the river. The tilapia should not be bred in any situation, any facility near to the river. Always the breeding facility should be in the ponds, not allowed to do in the cages where it can create a disaster. Now, tilapia industry, while smaller amounts were reported in West Bengal, Gujarat, Kerala, but as far as I see, the total production of tilapia in India is 18,000 tons only. And in Ghana, which is having 2.6 crores of population, here we are producing nearly 60,000 tons of tilapia. And that is all for the domestic consumption. And in India, hatcheries are mostly privately owned and operated independently from farms. Hatcheries operate seasonally with most production coming from the spring and summer. Sex reversed male tilapia fingerlings are typically sold for like 0.5 cents to 6 cents. Yes, in India, tilapia fingerlings price is extremely high when compared to the other countries, but it is a good opportunity for people who wants to venture into the hatchery of tilapia also. And major feed tilapia producers include Growell, Avanti, and Godrej. And here I would thank Growell to pioneered this tilapia industry and taking the responsibility and burden of the tilapia industry on their name and taking ahead as a brand. And nursery rearing is practiced at grow-out farms in small dedicated ponds stocked at 50 to 60 per meter square. And after 1 month, they are transferring to grow-out ponds. And there are examples of dedicated nurseries tank using raceway-based RAS systems and 120 per square meter. This data, I brought it from a research paper presented. And here they mentioned that grow-out ponds are stocked at 10 to 12 juveniles per meter square and fish is harvested at 400 to 600 gram size. This is the place where the people get misleaded. Like if I'm stocking suppose we stock 10 per square meter. And if I'm harvesting at 500 grams, that means from 1 hectare, I'm producing 50 tons of tilapia. Is that really possible? It's not possible. With this such misleading data only, the entrepreneurs enter into this business and they lose money. Here in Ghana, with an accessory aeration, we are struggling to produce 10 tons per hectare. The DO levels doesn't support so easily. We are also at mean sea level. We are not at any high elevation. So the practical data is missing. People are just trying to become entrepreneurs by searching in Google and watching YouTube videos. Yes, they are the guides, but they are not the truths. Now, tilapia here, they also mentioned that 500 to 600 grams in 5 months. That is also not so easily possible. Tilapia in cages are harvested with hand nets. Farmers mostly harvest their own ponds, pack the fish in ice and transport to local markets in open trucks. Yes. Now the markets for tilapia are all domestic. Some consumption is in the local producing villages while the balance is exported to the major cities. And the fish is delivered to the vendor who conducts the retail sale of fish. The fish vendor will pay cash for delivered at $1.86 a kilo. This is also a wrong information. Nobody pays $1.86, which is what we are talking like 130 rupees. And they also mentioned like it goes to 2.8 kilo in the last place. These are all misleading data. In India, no buyer is buying tilapia more than 65 rupees a kilo. No, not at all. And that too because of the demand of a tilapia in Kerala, that price is getting dragged. If it is in Andhra, they are not ready to buy tilapia for 30 rupees per kilo also. The survey conducted in early 2017 provided information that 18,000 to 22,000 tons are produced in India. Now, what are the entrepreneurship options in tilapia and the trends? Like what can we do? Tilapia farming mainly consists of 3 subsectors which opens the doors for entrepreneurs. One is tilapia hatchery, which involves breeding of tilapia, producing fingerlings and supply to the farmers. And with a budget of 25 lakhs, a 5 million fingerling capacity hatchery can be started. And the payback period will be just 1 year and the gross profit will be nearly 50 lakhs per annum. So for anybody, a fisheries graduate who wants to become an entrepreneur, he can look into the tilapia hatchery as a best option. Then we have tilapia growth, which can be done in ponds, cages, RAS units, and where the pond management of feed, high quality monosex fingerlings, low protein, high digestibility feeds, these all play a major role in the success of that tilapia growth. And this is also a major area where a fisheries graduate can think about. You have 10 cages for a tilapia growth, you need not do job anywhere. Like you can sustain yourself, you can live like a king. Then it's tilapia processing, where procurement of tilapia from wild catch or from farms, and it can be processed into frozen tilapia, whole round frozen tilapia for African markets, and it can be filleted and exported as frozen fillets or fresh fillets to Europe and American markets. There is one company in Indonesia called Regal Springs, and they are producing tilapia in Indonesia, Honduras, and Mexico. And they are producing 1 million metric tons of tilapia by their own. Only one company is producing 1 million metric tons of tilapia. So India, with its vast potential of water bodies and land, how much can we produce? What should be our impact on the global market? Now, tilapia hatchery technology, when I go into, tilapia hatchery technology has been developed in Asia and was followed by all over other world and other countries. And the various methods of tilapia, which tilapia hatchery to be followed are, first one is the Hapa methodology. This can be easily started. What we need is only a pond and some Hapas, where breeding Hapa, sex reversal Hapa, fingerling rearing Hapas, and all these Hapas are there. And I've been mentioning about the dimensions of those Hapas also. So we need the breeding Hapas where we stock our male and female fishes at the rate of 3:1 ratio. And we wait for 15 days and they start giving the fry. We collect the fry, we transfer to the sex reversal happas, feed them with the hormone feed for 21 days. And then we get the sex reversed fingerlings, we take them to fingerling happa, and again we feed them for another 15 days. We get 1 gram size or 2 gram size fingerlings, which we can sell to the farmers. Unfortunately, in India, all the tilapia fingerlings are sold at 0.3 grams to 0.5 grams, which is not that good. It doesn't give a much choice to the farmer. 0.3 grams at 2.5 rupees a fingerling, which is a very, very costly deal for a farmer. And like, you know, we get all the inputs which are needed for a tilapia monosex hatchery. The methyl testosterone we get in India, we can import from Thailand. We get alcohol, everything is available in India. It's nothing like, you know, we don't get something, we can't do this. Everything is available in India. And fry will be stocked at 0.008 or 0.1 gram size. And after 21 days, we get at 0.2 to 0.5 gram size for the sex reversal. And that will be grown for another 15 days to come to 1 to 2 gram size. And this is my tilapia hatchery in Ghana. And these happas, what you see are the breeding happas. And these happas, which you see in the next slide, these are all sex reversal happas and fingerling happas. And here we do sex reverse the fish and then fingerlings, and then we transfer to the fingerling happas here, and then they will be stocked in the ponds. Now the second hatchery method, egg incubation method. The second methodology of hatchery practiced is egg incubation method. And this methodology, the only difference is from the happa methodology is like here we collect eggs instead of fry from the mothers. And that egg will be incubated in the incubation jars and then incubation trays. And then they will be brought to the sex reversal, back to the same sex reversal happas. And then they will be converted and they will be grown up to fingerlings and they will be stocked in the ponds. So the only difference between egg incubation and the fry collection is the incubation of the eggs. And this is the typical egg incubation hatchery where you can see the egg incubation jars here. This is a FRP jar here, and they are the egg incubation trays here. And this facility is in Thailand, where they produce 200 million fingerlings of tilapia in a year. Now we will come to the third system of the hatchery called RAS system. The third methodology of hatchery practiced is RAS, and it's a typical system where we have the cement tanks, greenhouse, and where we will be holding the broodstock in the happas in the cement tanks. Again, fry will be collected, incubated. Then again, we take the fry to the FRP tanks or HDPE tanks, and again we'll sex reverse them there, grow them to fingerlings. This is all in an indoor environment in cement tanks or HDPE tanks. This RAS hatchery is very, very costly. It is almost 10 times costlier than a normal hatchery. High investment, high technique oriented, and high risk oriented also. And this is a typical RAS hatchery where you can see the greenhouse here, where you can see the cement tanks where the broodstock is held. And these are the HDPE tanks where the sex reversal is done. And these are the fingerling tanks, FRP tanks, and these are the biofilters. Like, what should be the goals of a tilapia hatchery? A tilapia hatchery management is all to produce quality tilapia fingerlings, which has to perform in the grow-out ponds or cages under certain traits, like, you know, metrics. We call them as metrics. Like if I stock 1 gram size tilapia, within 6 months, it has to grow to 400 grams, 500 grams. These are the metrics. Like I need to assure my buyer of the fingerlings to that, you know, my fingerlings perform like this. It will perform these metrics. So this is the critical part for a hatchery guy to establish his own metrics. And performance of broodstock in hatchery is recorded on the main parameters like fecundity, which can be the egg or fry, egg hatching rate. Like, you know, some hatcheries, the egg hatching rate is very poor. Some hatcheries who do manage well, their egg hatching rate is very good. And the survival rate of the fry during sex reversal, fingerling survival, growth rates in sex reversal, all these are the performance metrics of the fingerlings. So various authors and institutes have done a lot of trials on broodstock handling methods, simultaneously the fecundity rates, survival rates. In general, the scientific community prefers egg collection and egg incubation methods rather than the fry incubation and fry collection. This is all because, like, you know, when we relate to the science, it's the survival for the fittest technique. When we do the egg incubation method, all the trash will get, you know, they die. Only the strong ones survive, and they do perform very good in the grow-out. So we have considered all the recommendations by various authors and tried to implement as much as we can in our production methodology, and later started adaptive technologies which can suit African infrastructure, manpower capability, and stress-free management. like as I said, as every community has been recommending this egg incubation method, I also started the same. But the problem here in Africa, we don't have the infrastructure in a well-established way. The power is not continuous, the manpower is not trained. So we have been facing so many problems. Then I thought, okay, this is not like what I want to do is not what they should do. I should know what is they're capable to do, and I have to establish and adapt the system in that way. So we came back to the HAPA methodology, which gave a peace of mind and sleep to me. And we collect a lot of data in the hatchery to analyze our fingerling, quality and also performance, especially like, average fry production rate, sex reversal survival rate, fingerling survival rate, growth survival rate, average body weight index, average selling price of what we achieve by stocking my fingerlings. Like that, we do a lot of analysis on the fingerling performance. And we also do a lot of data analysis, like you can see here, what we have recorded the fry which we collected from a particular brood stock in different collections, and what is the average fecundity each time, and what is the average fecundity per month. And then we compared with the average fecundity of egg incubation method, and also in the RAS system. And here it is, like when I do the direct fry collection, I'm getting nearly 1300 fry per female, where if I do the egg collection, I'm getting only 600. And when I do that egg collection in the RAS unit, I'm getting only 500. And again, if I do the sex reversal survival rate analysis, the fry which I get for the fry collection, they are surviving only 50% in the sex reversal stage. And whereas the egg collection one is giving 60% survival, and the egg collection one is in the RAS unit is giving 80% survival. And in the same way, when we analyze their survival rate in the production system, the growth system, they range from 80 to 100%. The fingerlings which we produce in the RAS, they are giving the highest performance in the survival and also in the growth rate. Again, as I said, the genetic rule, the survival for the fittest is the reason behind this performance. And now when we go to the data analysis, the average body weight index, ABWI, for the fish grown in RAS method fingerlings is higher than the egg incubated and then the fry. And if you look at a stipulated time of 6 months, the fingerlings which are grown in the RAS unit, they are growing to 425 grams or 460 grams against 425 in egg incubated, and again 380 by the direct fry collection. This is all the genetic principle again. And we thought we also tried grading these fingerlings for 6 times. The fry collection fingerlings we are graded for 6 times, and we calculated the growth performance of that against the RAS ones. We got the same growth there. So that means the selection of the fingerling should be rapid if you are doing a normal fry collection method. Now, the cost analysis, we have seen that the performance is better in high technology systems, average selling price is better. But when we look into the cost component of that one, when we look into the capital cost of a hatchery setup by using HAPA breeding and direct fry collection, it costs $30,000. Whereas the same setup of egg incubation method using HAPA breeding costs nearly $60,000. And when you want to set up that same capacity in a RAS unit, it costs nearly $130,000. So the more you go for the advanced system, your capital cost keeps going high. Now the cost analysis in the second way is like the direct cost, which will be almost each and every hatchery will have the same direct cost. It's all the cost to produce fingerlings, or the cost what one fingerlings cost is implying to produce 1 kilo of fish. So there is no much difference in that. And the third cost involved is the overhead cost, which constitutes like salaries, power bills, maintenance costs, fuel bills, which will be very high to run a RAS unit than egg incubation than in the fry collection method. Salaries paid to expats, like in Africa, like we need to hire expats to pay to run these high technology systems. So the overhead cost will be very, very high when we go for the high sophisticated systems. Now, I would like to conclude the hatchery technology. The main constraint in Africa is sophisticated and reliable supply of infrastructure, not only in Africa, even in India also. Most of the places where we are starting our aquaculture projects, we don't have a proper power supply, a consistent power supply. We'll be having a lot of power cuts, And if you like to go for a RAS unit, you need to have a generator and again a backup of that generator, because within half an hour, if the power is not restored, the whole hatchery will collapse for a RAS unit. So conclusion of my study suggests farmers not to hype the system looking at the Southeast Asian or American farmers. Let's have our own adaptive technology suitable for our environment, infrastructure, and people. Now, I would like to go to the grower sector of tilapia. And here, I would like to ask our question, what type of growth suits in India? What should we go? What should we go for? But the ponds or the cages or the RAS? Which one is the best? First, we will go analyze the pond culture system. This is the typical 100-acre farm which I constructed in Ghana. And if you see, all these small ponds are the nursery ponds and all these big ponds are the grow-out ponds. And I have divided 20% to nursery and 80% to the grow-out. And my intention of doing that is like use the overwintering technique and to maximize number of cycles from a particular pond. So my fingerlings will grow for 3 months in these nursery ponds. And after that, they will get transferred into these grow-out ponds, and they grow for another 3 months in this grow-out pond by the time they reach the marketable size. So instead of 1 crop or 2 crops from one pond, I'm almost making 3 crops in a year. Even some ponds we are making 4 crops in a year. So I have maximized the production capacity of my farm by just adapting the 2-tier technology here in Ghana. So size of the farm is 100 acres and culture system of tilapia is 2-tier pond culture system, and number of fish ponds are 20 nursery and 20 grow out. And nursery is 0.2 hectare size, grow out is 0.8 hectare size, and shrimp ponds we have 24 ponds and each one is 0.6 hectare. And stocking density of tilapia in nursery is 25 per square meter and grow out is 3 per square meter. And stocking density of shrimp, the tiger shrimp, is 15 per square meter. Stocking size of tilapia is 1 gram. Grow out period in nursery is 90 days and grow out period, transfer size from nursery is 100 to 140 grams. And grow out period in main ponds also in another 90 days, where we are targeting 500 grams, but we are able to achieve only 420, 440 grams. Our target FCR is 1:1.3. And now we have touched a lowest FCR of 1:1.1. Target cost of production before overhead cost is like $1.2, but now we have achieved $1 direct target cost of production before overhead cost. And target cost of production including overhead cost is $1.5, and now we have reached that also at $1.25. So target selling price was 2, and we are doing 2.4. And this farm construction has started in 12th December 2017, and the first stocking of nursery ponds is done on the 16th February of 2018. Just in 2 months, we have made the project to get viable for the stocking. And the first transfer has happened on 23rd May, and the first harvest has been done on 30th September 2018. So within 6 to 8 months, we are able to construct the farm, produce the fish, and even able to harvest that. And from the total tonnage harvest target from our 20 hectares is like 600 tons, which is like 200 tons per cycle, 10 tons per pond. And the total shrimp to be produced is 120 tons in 2 cycles. And the budget for this farm to attain self-sustainability is $1.5 million, which includes the capital cost, construction cost, operational cost, everything. And we achieved in that particular budget. And the total length of the dikes in our farm is like 12 kilometers, and the employment generated from this project is like 3 expats and 50 locals. Now let us analyze the pond culture cost analysis if we do in India. The capital cost, if I do a pond culture, 1 hectare pond unit will cost nearly 15 lakhs rupees, which includes the land, maybe like 10 lakhs, and construction another 5 lakhs. And minimum number of ponds for a commercial operation should be 10 ponds. And the total capex will be like, the capital expenditure will be like 1.5 crore. And the operational cost expected production from a pond like 1 hectare in a 2-tier system, where 20% nursery and 80% grow out, will be like 10 tons. I'm not mentioning about that a figure like 50 tons, what has been mentioned in the earlier research article. And the FCR, what I'm target is still 1.1, where I require 11 tons of feed to produce these 10 tons of fish. And the average feed price I've been considering as ₹40, which is almost like $530 a ton, which I still think this is at a higher side. We can even bring down the feed price to 28 to 30 rupees. It will be much profitable venture of going into tilapia. And here, if the feed cost is like at the 40 rupees, 4.4 lakhs, fingerlings required is 40,000, which is like 80,000 rupees. The total direct cost will be 5.2 lakhs, which is like 52 rupees to produce 1 kilo of fish. and average selling price rupees 75. I'm not even mentioning 100, I'm just mentioning 75. So gross profit from 10 ponds in 3 cycles in a year will be nearly 69 lakhs. So the payback period for this project will be 24 months, and it is having a much better ROI. Now let us analyze the cage culture system. These are the cages which we did it in Ghana. These are HDPE cages, which are very robust. Even if the cages got detached and they travel 10, 20 kilometers also, nothing will happen to the cages. And this is a short video where we are feeding our nursery, the fingerlings. And the guy who is feeding is also an alumni of Ratnagiri College, Mr. Mohnish Chowdhury. And he used to be our farm manager in those days, and now he is in Nigeria handling a very big tilapia hatchery operation. Now I would like to show the cage culture grow-out methodology, where my friend will be describing the methodology.
This is one of the farm in Ghana where Prio 320 from Indonesia is being used. So you can see the feed boys are feeding the fish in the morning, and the fish are so aggressive for the feed, and you can see the floating feed in cages. I'm going to show you a cage where you can see a lot of the feed is floating. The fish are consuming them very fast. Okay. And he's our diver, diving inside the cages for checking any damages to the nets. And he checks the nets every day in the morning. If there is a mortality, he removes them.
So if you look at the cage operations which I have shown, you can see how much care we have taken in the construction of the cages also. We have designed the cage in such a way that there will be no escape by jumping. And also we have a feed barrier net where we don't allow the feed to float away from the cages. And we have a bird net not to allow the birds to come and contaminate our cages. So it's my design, which I have started, which I have modified for 3-4 times and brought to that perfection. And initially we have been using a wooden plank there, and finally we came up to a HDPE plank there. And now when we analyze the cage culture cost analysis in India, a cage unit cost, which I'm talking about a 5 by 5 by 5 meter cage, can cost between 60,000 rupees to 2 lakhs in India. Why I'm mentioning 60,000 to 2 lakhs is like If you want to go with the GI pipes with the drums, it may end up with 60,000 to 80,000. And if you want to go for a very good cages made by Garware, strong cages, maybe it may cost up to 2 lakhs also. So the minimum number of cages for a commercial operation is 20 cages, where the total capex will be like 15 to 25 lakhs. And the operational cost for this 5x5, like, you need to produce 5 tons per cage, where the FCR will be minimum 1:1.4. So feed required is 7 tons. Average feed price, again, the same what we took earlier, 40 rupees a kilo. So the feed cost to produce 5 tons will be like 2.8 lakhs, and the fingerlings required and the fingerling cost will be like 30,000, where the total direct cost is coming to 62 rupees per kilo compared to the 52 rupees per kilo in the pond system. And the selling price I have taken the same. So here also it's not bad. Like if you look at the 20 cages, 2 cycles in a year, you can get a gross profit of 26 lakhs, and the investment is a little bit lesser than the pond system. So here also the ROI is very good and the payback period is less than 24 months. Now let us go and see the RAS unit, what it looks like. And this is a RAS unit made with the cement steel tanks in Hyderabad by one of a famous farmer called Viswanath Raju. This RAS system has been developed, improved by Mr. Viswanath Raju. Like, he patents it. And it's a very good technology which he developed in steel tanks, which are non-corrosive with a high life. And you can see the water quality, how he has been maintained, like, drinking water quality. So that is the best RAS unit which I came across in India. And the critical components of RAS unit is like, fish tanks, mechanical filter, biofilter, degasser, oxygen enrichment, and UV disinfection. So the advantage of RAS is like when we follow whole system, it is a highly biosecured project of aquaculture. But when we go into the cost analysis of RAS culture, one RAS culture unit cost, like one tank costs nearly 15 lakhs rupees. And the number of units required for a commercial operation, like 36 tanks, where we can use 27 for grow-out and 9 for nursery, the total capex will be nearly 5.4 crores. And the cost of production will be definitely high. And if you look at the way, like, the same selling price, what we sell in pond or cages, this project will be never viable, because even 10 years also we cannot recover our capital investment. So definitely it should be in a place where you can sell the fish for a very higher price. Now, let us see how to decide a culture system. Like if I'm going to do a project in a rural area where the land is at an affordable price, definitely we have to choose the pond culture. And if you are in an area where the land cost is high, but the water bodies are available, you choose a cage culture. And if you are in an urban area where the space is a restriction or a climate is a restriction, or where you get a better value for live fish, that means somebody is ready to pay 200 rupees per kilo of live fish, there you go for a recirculated aquaculture unit. And a greenhouse, which is, if you want to do in a cold area, you go for a greenhouse and do a recirculated aquaculture unit. So the location, the market, the demand has to decide which type of aquaculture system we need to go. Now, again, the overhead cost will be very, very high, very, very low in pond culture, risk very less, disease can be treated or disinfected. Whereas we go to cages, the manpower cost and training cost, especially the divers, will be there. And the risk also will be there at the time of floods and also the disease outbreak. So the overhead cost will be high in recirculated system for electricity, but the manpower cost will be low because most of the recirculated systems are automated, run with only 1 or 2 people. So we need to choose based on the suitability, but not as per the government funding or subsidy. Because I have seen many cage projects, they have started just on the name of getting subsidy from the government, and they install the cage units. After installing the cage units, they will think which species I need to culture. And then they are forced to culture tilapia and pangasius, which our people doesn't prefer to eat. And finally, they will be selling that fish for losses. And within 2 cycles, like they are closed. The project will be shut down. So these things should not happen. First of all, you need to know why I am putting cages, what to grow. So now, how to design an aquaculture business unit? First job, to study the market preference and price of the fish in the market. Like now, I want to grow something. Why should I have to grow some fish which is having a good market value, 100 rupees, 120 rupees, 150 rupees. I cannot grow a fish which is having 20 rupees or 30 rupees a kilo. So the market preference, like in Andhra, people eat only carps. They don't like tilapia even today. They eat marals. So if at all, if I want to produce, I have to produce those species, not something else, and force the people to eat. Now the second job, select the fish which has the high market demand and study the culture methodology, culture period, and cost involved, both capital and operational. Now the third job is the selection of the grow-out method, which will be like a pond, cage, or RAS. After all, we all need an affordable protein, but not fish at $3 to 4 per kilo. So the best method is going for ponds where we use the green water culture, low FCR, low cost of production, high scope for employment, low chances of theft. And here the environmental conditions like temperature, cyclones, predators in water bodies also have to be considered before deciding the culture system. Now the fourth job, preparing a proper business plan, a detailed project report, and cash flows and budget. So without having a business plan, without having a DPR, without having the cash flows required, if you suddenly jump into the project, then you will start looking for the loans, debts, financing, which will never allow you to grow. And moreover, it will make you to collapse and sell other properties for this particular project. So the fifth job is like designing culture cycles. Suppose like tilapia takes 6 months to grow, your culture unit should be in a multiples of 6, so that we harvest every month, we stock every month. The cash flows should be comfortable for us. So planning a culture design is also a very, very critical for an easy thing. Like if you stock all your 20 ponds or 20 cages once, again you harvest all 20 ponds or 20 cages at once, either you will be having a higher price or either you will be having a lower price. Either you will be hit with a disease at once or like that. But when you do planning in a staggered way, every month harvest, every month stocking, first thing, you can sustain your buyers. Every month they are having a reliable supply of fish from you. So it's a multiple advantage by going the staggered stocking system. Now, what are the key factors of success in fish farming? Good source of quality fingerlings, good feed, and a very good technical farm management. And we have solutions and suggestions for every step of the way. A good source of fingerlings and good handling practices. Attributes of good quality finger. What is good quality fingerlings? Like the parent shore should be good, and the genetic center also we should know from what is the genetics of that particular fingerlings, where the parents came from, which generation. And the parents' size and age also matters when it comes to tilapia. If the brooders are more than 300 gram size, that particular the fingerlings coming from a mother of more than 300 gram size will not do better like what between 80 and 300 gram size will do. Or direct fry, we need to do all those things. Uniformity, size, aggressive attitude to feeding, no wounds, no fungal infection, no parasites on the body, no fin rods, no tail rods. All these are the attributes for a good quality fingerlings which we need to check for. And also, stocking to be done only in cool climate, that is like in the early morning or late evening. And fingerlings weight should be never above 500 grams per bag with minimum 20 liters of water at any size. So these all things has to be done. Now, metrics of good quality fingerlings, like how it grows. If I stock 2 gram size, within 200 days, it will reach 400 grams. And what will be the FCR? What will be the cost? All those things we need to be very, very clear before starting the operations. And good feed with better FCR and growth. What is good feed? Like good feed is not like with high protein. Good feed is not like a cheap price. Good feed is the feed which gives us a good returns. Like the cost to produce 1 kilo of fish. What is the feed cost to produce 1 kilo of the fish? Whichever is the lowest, that is the good feed. FCR might be 1 or 1.5 or 2, but I need an economical cost of production. That is a good feed. So you can see the good growth, good survival, which comes from good feeds and good FCR also. But ultimately, the economics matters a lot because we need to be ending up in a successful business. And the technical farm management, like what is the technical farm management? What is the importance in that farm management to get a profits? All lies here only. Like aquaculture is not just buying fingerlings from wherever we like or our friends recommend and stocking in our cage, buying feed whichever is cheaper and feeding as per the manufacturer recommendation, harvesting without any regular sampling and growth analysis, and finally blaming fingerling supplier, feed supplier, friends, and cursing ourselves for losing money. That's not the aquaculture technical management. You need to have everything as an SOP on your paper. And that we have to follow the SOP, then we will never fumble in our culture. So the technical management is to source and buy proper quality fingerlings, to track the mortalities and stocking and estimate survival in the cage and feed accordingly, to select good performing feed and feed them appropriately in protein and pellet size, to calculate break-even economics, profit and loss before harvesting or by conducting regular sampling analysis. And our secret of success in Africa, which is like techniques, we are the only commercial pond culture production facility in Ghana, in fact, in West Africa, when it comes to tilapia. We have brought down our FCR to 1:1.1 from 1.7 by selecting best feeds, best feed management, improved genetics, like we are having now F2 generation on our farm, and best protein management. Like nowadays, it's not the amount of protein in your feed, it's the digestibility protein in your feed decides your profit. So we use overwintering technique of our fingerlings in hatchery and again in nursery for the compensatory growth technique. 2-tier, 3-tier culture system to have minimum productivity from a unit system. We have broke down the culture into multi-levels and getting maximum number of cycles of productivity. Establishing our own value chain, direct sales, YAP-based sales, and the lowest overhead cost management. Our 100-acre farm runs with only 2 expatriates now. And this is our distribution reefer truck. So we have eliminated the farm buyers and we are bringing our fish directly to the city markets, and we are attracting the buyers there, and we have increased our selling price by nearly $0.3 by eliminating the buyers coming to our farm and we directly coming to the market. So now Mansfi Fisheries, which is our the company which I started for India, we would like to invest in Kerala, Maharashtra to build a hatchery, pond culture system and value chain. And we would like to produce monosex tilapia fingerlings using methyl testosterone hormone in larval feeds. And the hatchery will be very simple to start based on HAPA system. And to begin with, we will be like that, and maybe we will be developing into much high technique system. But we want to keep the investment at low and giving best quality. And at this 2-gram size fingerlings will be sold to the farmers or government agencies to stock natural water bodies, and also to entrepreneurs who would like to stock them in cages and other water bodies around. And my message is today, like US markets are looking for an alternative tilapia fillet supplier due to the trade war between US and China. India has the only potential to replace China in the fillet supplies to US. And tilapia price for China is not an index to export to US. They buy cheap from China, but in the same way they buy from Mexico and Brazil at a very premium price. Like FOB price in China is like $0.8, where the FOB price in Brazil is $2.2 and Mexico is also $2. So it's not, China price is not an index for us to measure and say, no, we cannot compete with China. No, our quality will be an index for us to get the market. So the quality matters and not just the price. We get all investment back to us in just one commercial batch sales for hatchery and less than 2 years for the grow, and it can become an income generation model for our fisheries graduates. So our slogan is give us land and water and we will give you fish and employment in less than a year. This is Khoon do main azadi dunga. Here I'm telling, give us land and water, we will give employment and fish. Thank you. Thank you for listening to me.
Very nice presentation, Nagamurli. And that your last slogan, तुम मुझे खून दो मैं तुम्हें आज़ादी दूंगा. तो तुम मुझे पानी और जमीन दो मैं तुम्हें fish दूंगा और employment दूंगा. And these are the 2 most important things. Like a fish, the protein for the poor and for the healthy, and so that the health is being managed or health is being maintained. And second most important thing, a challenge in India is giving employment opportunities, or how to create more and more employment opportunities with increasing population. So these are the 2 major challenges, and I hope tilapia culture will create tremendous opportunities in both having the fish feed, fish as a feed, and also having employment opportunities. You have covered in your lecture all the aspects of tilapia, tilapia hatchery, tilapia grow-out, tilapia processing, and almost all points related to tilapia you have touched. And I hope there are a few questions on that. In YouTube there are about more than 80 people now, and in Zoom about 32. The number is reduced because there was no range for geo in between. So once the geo is collapsed, then the problem of the connectivity. Okay, so thank you Nagamurli for very excellent presentation and your sincere efforts to bring the entrepreneurship among the students. Okay, when we have discussed last time, okay, we were talking about, see, why the students, they don't have the confidence to go for the entrepreneurship. Okay, and the one thing which they can, one thing which we were discussing was the communication. Okay, many times the students, they feel very low with respect to their communication. Many times they think that since they belong to some local language medium like Marathi medium or from the Hindi medium, so I want to tell all those students, those who are attending this particular webinar, Vaneshwar Singh is from Hindi medium. He started learning his English only after 10th, 11th and 12th. That was the English medium for him. So don't feel that if we do any mistake in speaking in a spoken English, no problem, go ahead. Okay, with respect to your entrepreneurship, okay, most important thing which Nagamurli has said, okay, that you must be able to prepare your own project. Okay, you must be able to calculate what shall be the investment and what shall be the returns on that. If you can calculate that one, I hope it will give you tremendous confidence. Okay, yes, I can do this. Okay, and that's the basics of the entrepreneurship. Okay, having the technical knowledge, no doubt the technical knowledge gives you confidence. At the same time, the capacity and eagerness to earn more money and more money will also go for the entrepreneurship. So Nagamurli, thank you very much for your fantastic presentation. You have done lots of hard work to come out with such a nice presentation involving all aspects of tilapia farming. Okay, then I think we'll go to the next lecture. We are celebrating 39th Foundation Day of College of Fisheries Ratnagiri, and we are very proud to have our 3 overseas students as our today's Foundation Day lecture series presenters. And 2 we have already heard. One, Dr. Baneshwar Singh. We have heard to the Baneshwar Singh. Then we have now, recently we have heard to the Nagamurli. Nagamurli has completed his talk. And now we'll go to the next and very important session by Amit More, preparing for a career in futuristic seafood industry. That's the topic Amit will be handling. And now I request Amit to please start your presentation. And I can see your background is our College of Fisheries Ratnagiri. And on that background, I think you are presenting your research. Please unmute your mic and please start your presentation. Amit.
Yes, sir. Good afternoon, everybody. I think what an excellent presentation by Nagamurli. Absolutely fascinated and floored by the amount of work that he has done and the amount of effort that he has put into aquaculture, fish farming, and his entrepreneurial abilities and skills to expand laterally and vertically in that space is pretty astounding. And I think it is a great opportunity for our students to learn how aquaculture is going on around the world. I mean, especially in Africa, and then he's expanding in India and so on. I think there are tremendous opportunities all across the space, especially in Africa, India, Asia, and have tremendous capabilities to export products all around the world. And I think this is the right time for our students to enter into entrepreneurial ventures and try to train themselves to try become something bigger, better than what some of your predecessors were, some of your previous students were. Not that jobs are wrong, jobs are actually right. They are a great tool and a great platform for you to learn something more, to get some more information, to get hands-on information. It's basically like learning to drive a car or a bike or a scooter using your friend's car. Okay, because if you are actually using your own and you go and hit it somewhere, obviously you're at a loss, but versus like you're at least with a friend, the friend is teaching you and so on, and so you have some guidance, and after that is done, then you go ahead and start, you go probably and buy your own car, scooter, or bike, whatever that is. And that actually is the same transition that I followed. That is, and there are other people who have followed the same thing as well, because we know the ropes. It's basically called learning the ropes, or trading the waters, whatever you want to call it. But you have learned how to do it, and then you are going to progress and start making money for yourself, start becoming self-independent, and start creating jobs. I think yesterday we were having a mock practice session, and Dr. Chaudhary said a very, very important thing. Instead of being job seekers, we want our students from the Fisheries College to be job creators. And I think that's a fantastic way to look at things. And that's why I want to talk about how you want to build a career in the futuristic seafood industry. And when I say career, it leads towards entrepreneurship and how you can train yourself as well. And I'll give some of the experiences that I have encountered in my life, especially with my startups and with my efforts as well. Personally speaking, I'm from Maharashtra, basically from all over the place. My dad had a moving job, so I had the luxury and privilege to grow up all the way from Buldhana to Jargaon to Alibag to Ratnagiri and Solapur, and I've been there and so on. And so I have seen this multicultural, multifaceted society. After finishing my undergrad in fish and fishery sciences in our Fisheries College at Ratnagiri, I moved on to get my master's at Mangalore Fisheries College in industrial fishery technology. Did a very interesting project to improve the surimi process and reduce the water usage while still maintaining the quality of the meat and so on. And I love processing. It is my heart and soul is in there. And one of the interesting and fun reasons that I tell people that I chose processing versus aquaculture is because I had a fish tank at home when I was a first year undergrad at Fisheries College. And sure enough, in like 6 months, a bunch of fish died. And I was like, I don't really think that I'm a fish guy, a live fish guy. And I was like, I think I'm going to switch to processing. It's much more fascinating, lots of machines, lots of technology, lots of interesting stuff to learn. And I switched. So went for master's in fishery sciences at Mangalore Fisheries College. Then I proceeded to do another master's, and at University of Alaska, Fairbanks, and I was living on a small little island called Kodiak Island in the middle of the ocean and studying seafood spoilage microbiology, which was fascinating in itself because you're living on an island, and we had a tinier island next to it, and we were studying salmon spoilage, we were doing lipid biochemistry, we were doing microbial lipid chemistry, so on and so forth. So it gave me a tremendous good understanding of the seafood industry and seafood processing in Alaska and the fish and the environment over there as well. And then moving down south, I was like, I want to learn more. I want to be an interdisciplinary scientist. I wanted to be a scientist to begin with anyways, but I wanted to have an interdisciplinary approach to science. I wanted to learn what are the other people doing, because what happens most of the times is that we are in our same world. We are doing seafood, and we forget that there are other processing industries. There is there is poultry, there is beef, there is pork, there is produce industry, there are food engineered food companies, like say a chocolate bar or candy bar or chips and stuff like that. So there are all these industries, and they have all different kinds of techniques, technologies. The biochemistries are different, the microbiologies are different, but the fundamentals remain the same. And somebody mentioned earlier about the fundamentals. It is the fundamentals that I learned at Fisheries College Ratnagiri that are helping me right now. I mean, I can tell you multiple examples. I mean, talking from engineering department, we had engineering courses, navigation and seamanship. People don't know what navigation and seamanship is. I mean, people have hard time. I was at a very, very upscale museum in Los Angeles, and they had a horizontal sextant there. And it was just, somebody was selling that one, as a museum art piece. And I said, I asked that guy, I was like, do you know what this is? And he's like, yeah, it's horizontal sextant. And I'm like, do you know how to use a horizontal sextant? And he's like, no, I don't know. But I knew about horizontal sextant. I knew, I'm not only there, but I know about the aquaculture ponds and how to give feed and so on and so forth. And now I'm a seafood guy. I understand aquaculture. I'm in the poultry business now, and I'll get into that story in a little bit. But all these experiences kind of helped me to become a stronger scientist, and then cross-adapt and cross-apply different principles and practices that I have seen and learned in these different courses in our college. It could be a fish biochemistry course. Dr. Pagarkar was teaching us that, and I was a horrible student. I mean, I'm telling you right now, Naga Murali, gold medalist, and Baneshwar Singh is a gold medalist. I was an average student, 7.7 GPA, but it was not for personally speaking, I was there for the knowledge. GPA didn't matter much, because I know as a fact that I have so many friends who don't have greatest GPAs, but they are the best entrepreneurs. And you have probably met them. Bhupendra Parab doesn't have the best GPA in the world. He is not an 8.4. He is not 8.5, something like that. He would be somewhere about like 7.2, 7.5, and so on. But he is one of the best entrepreneurs there is. Sachin Surve, who is the manager of Gadre Seafood Processing Plant, he does not have a great GPA. So don't beat yourself if you are not having a greatest GPA in the world, but you can make things happen if you have the drive, you have the passion, and you have the knowledge. So when you are attending classes, go for the knowledge. And one of the best things about Fisheries College Ratnagiri is the fact that you get knowledge. You go out there on fishing trips, you go out, and with Pause sir, you walk the shores and you collect the samples, the mollusks, and you're collecting the crustacea and the mollusks and so on and so forth. It's so fantastic. You're not just rote learning in the class, but you're actually experiencing that. And that's why I would recommend you guys to experience the science that you're learning in the classes. I try to teach that same thing in my classes at where I'm teaching right now as well. So I wanted to get an interdisciplinary experience. So I switched fields to poultry science, which fortunately I was doing microbiology. And right now I'm doing microbiology, product quality. So again, the fundamentals remain the same. The proteins remain the same. Gelation, gel forming capabilities, products, farm products and fried products remain the same. But fish still has a lot more diversity. You have 300 different kinds of, and just as an example, 300 different kinds of commercially available species, and yet the amount and the number of different variety of products that are available in the market might be slightly lesser. Poultry is one species, and we have tremendous amount of further processed products, food products that are out there that are loved by people. I also want to, I just saw that Dr. Swapnaja Mohite came online. I want to tell her a big, big, big thank you. The reason why I went to see Fisheries College Ratnagiri, and the reason why I'm here today is because of her. She told my mom, my mom met her and her son was in my mother's class. And she said, don't worry, let him take fisheries. There is a lot of scope. He will really enjoy it. And I totally believed in that. My mother and my father, everybody believed in those words. And today I am here because of those simple few words. So anyway, I don't want to take too much of your time, and I want to dive into the subject of building career into futuristic food industry. So let's talk about the future. This picture that you're seeing in the background is actually from a movie called Back to the Future. Okay, where a mad professor and his student kind of go back into 1955, from 1985 to 1955, and there is a time when they move forward as well. They go into the future as well. So I just want to take you to the future and then talk our way through. What is the future? But what is the current situation? That is something that you would like to know. This is the current situation. You've got all the millennials, okay? The millennials are the ones who are born between 1986 to early 90s, 1990s, 95s, and on, until 1995. So that 25 year period is where all these millennials are. And all of you guys who are out there, and probably young professionals, you are connected to the to the web, to the internet. You've got Spotify, you've got emails, you've got Facebook, you've got Instacart. You're doing all sorts of things. So this is your customer base. Remember this, that if you are ever trying to get into the market and you're trying to serve the market, that is what an entrepreneur does. An entrepreneur has to find an opportunity to serve the market. If you have to serve the market, you need to know the market and who your customers are and what they're doing, what they are liking, what they're disliking, what are their belief systems, and so on. So millennials are obviously having cell phones, smartphones. They are connected to the internet. They're constantly talking to each other. And these are the customers who are potentially in developed countries, all over the globe. And so they are constantly chatting. They're sending pictures, they're sending texts. So if you go in a restaurant, you're taking a picture and uploading it and saying, hey, by the way, eat over here because this food is great. Or you're eating some fish and saying, wow, this is the greatest tilapia ever, or this is the best salmon ever. And then suddenly it goes on Twitter, it goes on Instagram, and everybody knows, hey, salmon's great because person X told that. And person X, why are you listening to person X? Because you have faith and belief in that system, and you have trust in that system, at that particular person. So suddenly now everybody is trying to go for salmon. You're going to go for tilapia, you're going to go for crabs, or whatever that might be, right? So understanding the customers, where they are, what is the space they are living in, becomes very, very critical, because that is the data that you kind of get and then start trading. Maybe you're catering to these guys and you want to set up an online system for ordering food and delivering food, like several of those that are actually in India, right? But this is one of the target populations, but these are older people. Now it is, forget millennials, it is about Generation Z. Generation Z is the ones who were born from 1995 onwards. So 1995 to 20, it just feels like I'm getting older and older by the day, but it is the Gen Zs, and probably some of you are in the Gen Z population who were born probably after 1995. And this kind of a population is all over the world. They are the first true digital natives. They were born, you guys were born in digital India. You were born in this digital age. You are seeing and adopting this kind of technology as well. I mean, when I was born and I was growing up, no telephones, one TV maybe in the entire area. I used to live in Jargaon, and Jargaon did not even have like proper roads at that time, so these kind of things. But you guys are different. You guys are at a whole another level. And this Gen Z population, who is also your potential customer, they believe in things. They believe that if you want to work for a corporation, they want to buy food, they want to see the social responsibility. They want to see the corporate responsibility. What are the values of the company? What are the culture of that company? What is the product? They're just not buying a product. They're buying the culture behind the product. So if you guys are into aquaculture, you want to show these Gen Zs, because they are going to pay the top dollars for your product. You want to show them that you are no antibiotic free, or no antibiotic ever, or antibiotic free product that you're selling into the market. Is it a organic product? It is a no preservative ever added product? Or are you is your company donating to a social cause? Are you helping a local population? Are you employing local population there? And that actually brings in a socio-economic change in that particular region. So Naga Murali was telling about his aquaculture farms, and he is employing those people. So these kind of things that are making socio-economic changes in your society are very important to these Gen Zs. What you need to do is that you need to start marketing those kind of attributes, those kind of characteristics to this generation. Okay. And when you are talking about entrepreneurship, if you are getting into aquaculture, yes, you want to start with 5 acres, and that is great. But if you are starting with 5 acres, you want to then start incorporating these values, incorporating these cultures, and then market them to the right people at the right time to get the right cost for your product. And there are people, and I can guarantee you, there are people in the US, there are people in Europe and all over the developed world, developing world, and even in India now, where people are paying premium dollars for these kind of cultures, like local food. For example, hyper local is another concept that is happening in Mumbai, where one particular society will somebody in that one particular society will be growing crops, or like growing vegetables and stuff like that, and selling it in their building, and it is an extremely expensive product. Of course, is it sustainable? Is it highly profitable? Probably not. But if you are an entrepreneur and you start multiplying that model in multiple societies in Mumbai, housing societies I'm talking about in Mumbai, you're probably going to make money. So an entrepreneur is always thinking about money through serving the population. And I also wanted to, so Gen Z is the population that you have to target, because they are the ones who are going to have the monies to spend in the future if you are going to be an entrepreneur. And so with that being said, this Gen Z, the millennials, all of them are actually going on for online shopping. You must have seen the rise and boom in online shopping with this COVID-19 outbreak. Everybody is on online, and they're ordering stuff. Amazon is in India. You've got Walmart is in India, obviously, and Walmart probably will start an online delivery system as well. Well, Amazon and Reliance is putting together a coalition for online delivery. So this is going to happen. But there is an online delivery. Instead of just buying toys and clothes and shoes online, people are buying groceries online on a regular basis. You go online, you type in what you need, you pay using your credit card, and the food is delivered in boxes. Now, right now in the COVID situation, me and my wife, and we have a little son who is 2 months old. We're not going into the grocery store to get the product, because we are afraid that we are going to contract the virus. So we are always doing this online service, and the food is actually, they bring them in bags and put them directly in our car in their parking spaces, and they put them in the car. And that is fantastic, because I have the ways and means to approach the grocery store. It's a small town. I can do it. And more and more people are doing online groceries. Okay. But on top of that, there is another concept, which is, as you may see in this box, meal kit delivery system. So the millennials and the Gen Zs are actually not inclined to cook at home as much. Okay. In India, there is a slight shift in the similar direction, especially in cities where women are working as well, and as well as their husbands are working. So they really have very less time to work, time to make food at home. And they want something that is fast to cook, good to eat. But the previous generation, who was the fast food generation, that's gone right now. It is this new generation, like your guys' generation. You want to cook food, but you want it to be cooked at home. You want it nutritious. You want it healthy, and you want the product to be just phenomenal. And that's why companies in the US, there are companies like Blue Apron, as a matter of fact, and there are several others, Tasty and so on. So what they do is they will take 3 recipes, okay, and they will create a bag, basically, of all the ingredients that are required for that each recipe. Say, for example, in this case, if you look at this picture, you can see a salmon there. So it is a pre-cut, pre-portioned piece of salmon in a styrofoam tray. It is beautifully packed. And then you have all the ingredients that are required to make that salmon dish that will go in that one bag. Okay, and it is sometimes you will get an onion, you will get a little bit of garlic, you will get some spices, they will even send salt if it's needed, and they will send a recipe. If you follow the 4 simple steps in that direction, you should get the exact recipe that they have listed on their in that pamphlet that is in that box. And they will send you 3 recipes. Each recipe makes food for 2 people. Now remember, we are, as a society, we are moving towards nuclear families, where family has maybe 1 or 2 2 people, or maybe husband, wife, and a kid. Okay, so these nuclear families don't need a lot of food, but they need time. They need time to spend with their families, with themselves, actually. And so time is becoming important. So we are giving prominence towards convenience. But where does that lead us to? Okay, one thing, if you are an entrepreneur, you are in India, you have got a big space to cover. You have a tremendous huge space to cover, and you can actually spread yourself in just getting the product, boxing it up, and shipping it to people. Right now, people are sending individual fish. They might be selling individual chicken and meats and stuff like that, but nobody, I've as far as I know, people are probably not doing these individual beautifully crafted, designed recipes that somebody can just take, put it together, and boom, you get a very tasty, healthy meal. Where does that lead us to as an industry, as seafood people as well? If you look at it, you have got a salmon fillet that is, and it looks like a farmed salmon, basically, although it says wild Alaskan. It has a very nice texture. It is pre-portioned in a smaller piece. The more you portion, the more you slice here, the more you fillet, you're exposing the surface to a lot more bacteria, temperature abuse. You've got a lot more moisture loss, and so on. Now the product quality starts to degrade. Okay, then this thing goes into a box with all this produce and everything like that, and it is getting shipped from one DC, which is a distribution center or their manufacturing site, all the way to your customer, and it takes about 48 hours, and it may sit at your doorstep for another say 6 to 8 hours. So the product quality has to be maintained, and so now you have to design a product, you have to farm, and you have to process a product that can withstand the supply chain rigor. And in doing so, if you are an entrepreneur, you may look at it and say, by the way, you know what, I can actually come up with a better tray that can hold the fish, or that could hold the chicken, or whatever that might be, that can absorb the water and can improve the shelf life of the fish or chicken during storage and transportation. I mean, there are these opportunities that you can take, and probably step 1 could be just trying to explore the market. And there are people who are over here. We have got in my town, there's a place called Hornsby Farms. Hornsby Farms, they have a farm, and a lot of us who come from the Vidarbha region or some other parts, and even in Konkan region, people have farms, and they grow vegetables and produce and stuff like that. This is the same kind of an operation, but what they do is they have subscription service online, and so people have subscribed, and they're like, every month, every week, we are going to send you fresh vegetables at home. And so farm picked vegetables. So they go in, they pick their farm picked vegetables, they put it in a box, whatever is available during that season. So now seasonality comes into picture, and that plays immediate role into the hands of the millennials and the Gen Zs. And so now they're shipping those boxes, very, very expensive boxes, but people still love to buy that local, fresh, from the plant grocery. So this is another business model that you can look at and incorporate seafood into it, or maybe you can make a pack of seafood, a box of different kinds of seafood. And that brings me to my next slide, which is called Alibag Seafood. And this is a company that I have partnered with in India, and we launched it recently. The company believes, and my partner is Rajaram Patil. Rajaram Patil is a fisherman. He comes from a fisherman community. He has multiple boats, trawlers and persimmers, him and his extended family, I should say. They have a dry fish operation. And Rajaram, unfortunately, although being from a fisherman community, he decided to move out of that and become a nuclear medicine expert. Okay, he's a nuclear medicine expert, has traveled the world. He came back to India, and he said, why am I not doing fish? He is now focusing 50% of his attention on actually selling fish than and 50% only on the nuclear medicine aspect. That is the power of fish. That is the power of seafood. That is the power that we have, and we need to explore, because he saw what is happening in the world, and now we are trying to replicate it in India, in Alibag. And the motive behind this is not to sell fish. The motive behind our company is to actually help the fishermen and to give exposure to the fishermen that are catching the fish, bringing them to the forefront, bringing the fish directly from, buying the fish directly from onboard fishing vessels, processing it in our small processing plant in Alibag, pre-packing it, and then selling it directly to our customers in Pune. So we have started this initiative in a very nice manner. Our hearts are with the community. Our hearts are in the betterment of the community, because now the fishermen are actually benefiting. Instead of selling a fish at a wholesale price, we are buying the fish at a premium price of 150, 200 rupees, or even more, depending on the quality and the size of the fish. But in return, what do we get? We get an end-to-end quality control. So it is called total quality management. And we have installed quality assurance parameters for onboard handling of the fish. So we are going to give them a box. We are going to give them ice, and we are going to say, when you catch surmai, you have to put that in this box, and you have to tell us what's going on. Through the mobile data, through other services, we will be able to know that they have caught the fish, and then they can share the data with us. Then we have a small processing plant where we are actually employing the local fisher ladies, the fish sellers from that particular community. Now the money suddenly goes to them as well in their houses. We are using traditional methods to cut the fish. Instead of having fillet knives and we are having big machines, we are actually using the same kind of the curved sickle like structure that those ladies use, so that we still are processing it in a traditional yet much more hygienic manner with a HACCP plan, with a GMP and everything like that. And the fish will be distributed, or is being distributed to our customers in Pune within 6 hours post catch. So it is that fast of a supply chain that we have maintained, and we are getting great reviews from our customers and our customer base. And again, I tell you, our customer base are the IT folks. Our customer base are all those guys with white collar jobs who are willing to pay a premium because they have got a premium product, and we are taking utmost care in terms of packaging, in terms of holding, in terms of cold chain management, end to end. Now, in doing so, we are also mentioning that we are going to give you, and so Rajaram being from the Kori community, from the Agri and Kori community, they make fish in a very different manner. And a lot of people in the cities would not know that, because the masala that is used, the spices that are used are completely different, or they're slightly different in the Agri Kori community. So now we are putting together recipes, we are putting together these masala packets and other things in the box, now replicating like the Blue Apron style things. And then you will directly get a box in your front, delivered to your doorsteps, and that person is going to actually get the food, get the fish. They are going to have all the product, and they are going to make the fish according to the recipe if they want to. Okay, so we are progressing in that direction already because of COVID situation, and it's a very interesting dynamics, because there were people in Pune who actually lost their jobs. They were salespeople and so on, and when they found out about our initiative, they said, why can't we become your distributors, co-distributors, if you will, sub-distributors in certain areas in Pune? So our driver doesn't have to go to each and every household. We are just going to dump that entire box or entire cooler full of products into this person's house or this person's facility, and from there they are just going to go in and deliver it to other people. I mean, this is the model that you start to explore. And when we are talking about the fisheries and the fishermen at the heart, we are actually working with the Sahkari societies, which we had learned in undergrad, by the way. Otherwise, I would have no idea how they function, what they are, and we have contact with them. And so can we replicate these models for inland fisheries, where they will have their local fish that they can box and they can ship to other parts of the world, because everything is online now? Okay, we are building our online platform, but this is how the concept begins and how we can get these entrepreneurs, get you guys to think in a novel manner. I mean, if you are interested and you're in different part of the country right now, and you want to replicate our model and want to be a franchisee owner of Alibag Seafood, we would be more than happy to talk to you about the model. But all it is about providing fresh seafood to the customer, with the focus on the fishermen and the people who are affected, who are in that particular community. And that appeals a lot more to the entrepreneurs, and we believe in those principles as well. So the next thing that I want to talk about is actually blockchain, a complete different change in topic, but we are talking about IT, we are talking about technology sectors, and we want to go into the next phase. And next, because I'm talking about futuristic technologies, future that you need to prepare yourself for. There is something interesting called blockchain technology, which is now upcoming and getting rapidly adopted across the globe. In fact, fishermen or aquaculturists in Andhra are actually going to use blockchain technology in conjunction with IBM, I believe. So what is blockchain? Blockchain is basically collecting the data and uploading it on so it gets uploaded on the cloud, if you will, in terms of like blocks. And as the fish goes from one step to the other, and let me just go to the next slide. As the fish is going from, once it caught, once it goes to the fisherman will collect the data, it will get uploaded as a block. Then that same fish goes to the middleman takes the temperatures, it gets uploaded as a block. And now and it goes to the factory, to the retail, to the customer, and whatever data is getting collected, it gets uploaded as a block. That block of data is then now connected from the fish all the way to the customer, as you can see in the diagram over here. So the data was collected for fish and at every interval, and now the customer, if they scan a QR code, scan a barcode, they will know exactly where the fish came from, what were the temperatures, what were the parameters. So now the authenticity of the fish is now approved. Now you know that there is credence. So the credence value of that of your operation increases tremendously. And blockchain is a very, very interesting and novel technology. The data that gets uploaded, it will be is immutable for most part. If you have to make a change, you will have to go through a series of steps, and you will have to actually create a new entry into that entire system. One of the interesting aspects of this is that the data otherwise, in other data systems, it stays in a central computer. Okay, and a few people have access to that central computer data system. Here, the data gets shared among different shareholders in that entire system, so they know what the data was entered by the fisherman. The customer will know what, or the retailer will know what the data was entered by the fisherman, and vice versa, and they will validate each other's data as well. So creating a very interesting, trustworthy network of fish from farm to fork. Okay, why am I telling you this? Because we were talking about aquaculture and aquaculture. It is great, but we need to have that credence. We have to have that provenance of our system. Say when you are talking, when Nagamurali is talking about the cage culture in tilapia over there, why can't we, or say Nilesh Shinde is talking about his shrimp farm in Raigad district, why can't we take the data for the fingerlings, or for the eggs, and where the hatchery is, and what is the lab report, and all that gets uploaded onto the blockchain. So each batch of that shrimp or the tilapia fingerlings, wherever it was procured, you have all the connecting points for that. How much antibiotic was given, or if it was not given, what was, some of the information could be private, some of the information could be publicly shared or with other stakeholders as well. So you can have that leverage. But what you have created is that if the customer wants to know more information, the retailer wants to know more information, at a click of a button, you can go all the way back to the fish, all the way back, and you can get all that important information that is needed for decision making in terms of selling the product, in terms of processing the product, in terms of marketing the product or positioning the product in the market as well. Okay, so blockchain has become a very, very important tool, or will become a very important tool in the near future. And let me tell you one thing that I forgot to mention in the previous slides, is that if you are a company, if you are an entrepreneur, you might be selling fish. You may say, you know, I'm an aquaculture company, I'm a seafood processor, I am something. You're not. You're actually a data company. Every company in the world is a data company. Remember that. So let me give you a quick example. There's an entrepreneur in the US who started a clothing store and a clothing business where she was making custom made dresses for people. So you give your dimensions and that lady would make the dress. Then she started, you know, having more tailors, and it is a tailor-made dress, right? But it's all online service. And the interviewer asked her, so you were always a clothing company? She said, I'm not a clothing company. I was a data company from day one, because you're collecting data. Now, in this case, and even in terms of Alibag Seafood, when we are going to collect data about the fish, where they are being caught, we know the location of the fish where they are being caught. We know how much the fish were caught. So now we know seasonality and how the fish are migrating. How awesome would that be? How awesome would that be for my customer if they were to look at the data and they were like, oh yeah, I know that the fish was caught here, or X or Y, wherever that is, you know. So the journey is important. And the customer, the millennials, the Gen Zs are interested in that. So that is your customer. If you want to make more money, you want to invest in this technology, you can actually do that. And some people say, well, who's going to pay for all this stuff, right? So IBM has Food Trust, and I'm not part of IBM, or they're not paying me, but IBM has Food Trust. And if only when the data gets used, somebody has to pay money for it, and it's generally the end user who pays for it, like McDonald's, for example. So this is a very, very important technique and technology that students should start learning. And there are several online learning courses that you should learn and become next level of entrepreneurs who understand fish, you understand seafood, who understand our business, and now you're joining it with technology. You would be one of the best ones out there, guaranteed. And I want to show you this particular slide. I'll be quick over here. This was a fenugreek outbreak that happened in Germany, and as you can see, the red dots in that diagram are where the outbreak happened, and people started dying, and they were not able to figure out what is happening. Why are people suddenly dying? 50 deaths, 4,000 people were sick, and it took them 40 days to figure it out as to where things were. Where is it coming from? Because the seeds traveled from Egypt through Antwerp, went to Germany, went to France, and started affecting people. So the supply chain was so complex, nobody knows where the food is coming from. And so in order to track the supply chain, in order to figure out where the food is coming from, you actually can use blockchain technology, and blockchain actually can trace a product in 2.2 seconds instead of 7 days. And this was an experiment done by Walmart. So it's a fast way to track a product, stop food safety outbreaks from happening. Then why is it important, and how is it important for our industry? Well, it is actually, you can store the data chronologically. You have rapid data acquisition, food safety, traceability, transparency. You've got a lot of marketing tools as well. And it can be used, and there are some hurdles. I mean, it's a new technology. You will have data authenticity problems. So something that you need to learn. Next thing that I want to talk about is data analytics. Data analytics is another big field that you want to learn. It is more statistics and mathematics and algorithms than you could think, but you need to combine those techniques. And what you can do with data analytics is you can use it as a prediction factor, where you can take farm management, environmental factors, pre-harvest food safety, and predict where the salmonella might show up. What is the shelf life of the product? Some of the things that we are doing is we are doing data analytics to predict active shelf life of the product, so we don't have to just rely on the shelf life that is printed on the packaging. So this is something that you guys as entrepreneurs can take up as well. Data is always collected in our processing plants, but it is never used. So if you are deboning, is it the deboning and you see this X-ray and there are some bones? Why is there a bone? Can you, as an entrepreneur and as a consultant, if you will, you can go in and troubleshoot these problems using data analytics as well. Another thing that we are working on is actually called augmented reality. And I may not want to go into detail into this, but it is basically like Google Glasses, where you can actually see what the other person is doing, and you can provide solution rapidly as well. So this could be used as a tool by entrepreneurs and consultants to tackle problems and troubleshoot problems in real time. But here, this is one thing that I would like to talk about much more in detail. It is called functional ice. Functional ice is something that we have developed. I have coined the term called functional ice. It is a modification over your regular ice. And it is basically adding certain ingredients, food grade ingredients or chemicals, if you will, into water, freezing it into ice. And what we have done is we have used that block of ice. This is a project we did it in Honduras. We use that block of ice on shrimp, and the lady is selling fish on the street side. So fish and shrimp. These shrimp were from aquaculture ponds in Choluteca. So as you may notice in this, as it was told by the lady who was selling the fish, the shrimp which were on functional ice stayed fresh for a longer period of time, over 8 hours. Those shrimp were absolutely fresh and they were turgid, versus when they were kept only on regular ice, the shell was kind of coming off. And you will notice one interesting thing is that the flies kind of went away from the shrimp and went and sat on the fish which were not treated with functional ice. So functional ice is a very interesting and a simple concept that we have developed. And as an entrepreneur, we are launching it in India. In Alibag, basically right now, and this is something that could replace formaldehyde. So we have a technology which is easy to use, easy to manufacture. You don't have additional infrastructure costs and everything like that, and you can still make things happen. And that is the beauty of innovation. That is the beauty of an entrepreneur, to come up with technologies that could be easily adopted, replace the existing model, and can benefit the end customer. The ice melted very slowly, so the ladies didn't have to spend as much money on ice either. And so it was a fantastic project. We're going forward and we are transferring that technology to other people over there as well. This is another project that we had done in Honduras, in Nigeria, and this is a testimonial by one of the users, and we are targeting the ladies. I may want to skip the video right now, but let me just show you the pictures here. So these are the ladies who are selling fish on the street side. The lady in the blue t-shirt and the lady in the center with the gold necklace. They are my collaborators in Nigeria. We are making functional ice and giving them in bags, and they have seen tremendous amount of difference in the way ice melts, and that is why their fish stays colder for a long period of time. The fish stays fresh for a longer period of time. What does that ultimately do to those people? It actually saves the fish from spoiling. That means they can sell the fish at a higher cost. At the end of the day, they make an extra dollar. That extra dollar goes to their family, to their kids who can eat more food, or they can go to school, or they can do whatever that is needed. But now that extra dollar helps that entire family. Now suddenly, by one intervention, you're helping the entire socioeconomics of that particular community. And the women population that we have targeted is actually, most of them are widows, or their husbands or children have gone to fight wars, or they are part of the pirates and stuff like that, because this part where we are doing the trials is heavily infested with pirates. And so helping these women will actually help the population there as well. Last but not the least, I want to, as an entrepreneur, you always look for problems, and you want to always think about how you want to solve the problems. The number one problem that we're having, which is coming up, we are going to have 9.1 billion people in the world, and we have to feed 70%. We have to increase our food production by 70%. That means you can do aquaculture, you can do seaweed culture, you can do the mollusk culture that we were part of, and I was very proud of that. But at the same time, we have a lot of loss or waste of food and fish. Okay, just to look at fish and seafood, 35% of the fish and seafood that is being produced in the world gets thrown away, and that's a very, very sad reality. If you can, even as an entrepreneur, as a young student who are looking at things, if you are finding one thing that, hey, can I make a difference over there? If you can, and you can save 2% of the fish that are being thrown away globally, it could mean billions and billions of dollars in bottom line. And that's what actually functional ice is supposed to do. The idea came to me, when through all the experiences through Fisheries College, walking through villages, talking to people, seeing the poverty in our fishermen community, as well as doing the work experience of that ice making plant near the MBRS. Okay, our environment is growing bad as well. So what are some of the things that we can do over there as an entrepreneur and at the same time have sustainable fishery there? Overfishing is happening as well. So what are some of the things? Are you looking at trash? Are you looking at byproducts, bycatches? Are you willing to convert that into some very interesting, important product that is feasible in the market? Not because chitin and chitosan has been dealt with for years and years. People have been, you just go in and type chitin and chitosan. You need to move on. You need to either take chitin and chitosan and make it into products that could be sold at an affordable and good price, or otherwise, just stop, because it is just overexploited chitin chitosan market. Maybe you can improve the process and all, and that's good, but you need to come up with viable products. Okay. But with that being said, I just wanted to inspire the entrepreneurs and students in our college. And with that being said, I'm just going to give these last few opportunities. You can do local fish. You can have value-added products that are from your region, and you can sell it in the market. Market in the sense e-commerce and so on. You can sell ready-to-eat fish or cooked products, or you can start your own fish food testing lab as masters and PhD students who have undergraduate or who have understanding of food testing as well, which is very important. Okay. With that being said, I'm just going to stop. This is my email address. I'd be more than happy to help you guys out in any way possible. Thank you very much. Naga? I think it's just both of us.
Yes. Yes.
Yeah, I mean, actually, I also have started the same type of app here in Ghana. As I shown in the beginning, Pukio Fish is my app name. And now I have opened the app for all over the world. And I would like to come to India that app very soon. This Hyderabad. So let's see how we can integrate.
Yeah. I'd love to talk. There are multiple things we have to actually collaborate, it looks like.
Tilapia, tiger shrimps. Along with that, I am sourcing the sea fish, like, grouper, snapper, kingfish. And we are supplying to our Indian community, especially during this COVID situation. For that reason only I started, but it is now getting developed into a better sales opportunity.
Yeah, that's exactly our story.
Yeah,
that's exactly our story. Just got started and it is growing. And so we are capturing some markets, but our heart is with the community, and we want their betterment as well. And so definitely willing to replicate it different places and all.
Reliable, fresh seafood at your doorstep. And that's our, this thing, we are doing a door delivery also with the delivery boys.
Yeah, exactly. Yeah, it is sea to kitchen. We are getting it from the fishermen and so on. No, good, very good.
So very nice presentation by Amit. Amit, I hope you can you hear me?
Yes, sir.
Yeah, my video is having some problem. So I think we are coming to end our program. Before that, I think we'll have some questions. In the chat, we are welcoming the questions. If somebody want to ask direct questions, please unmute your mic and you can ask your question.
So, Amit.
Yes, sir.
So how the functional ice is different from the real ice? What composition, like in terms of, its chemical structure or something? How
Sure.
it is different from the real ice?
So basically what we have done is we have taken a couple of food processing aids or even food ingredients that have proven antimicrobial activity, but at the same time, they have some other benefits. So functional ice is defined, and I have defined it as functional ice is an ice, is a modification or innovation over your regular ice that has antimicrobial properties, that melts slower and can improve quality. So we have chosen ingredients and chemicals that can actually do these multiple things at the same time. Not all the three boxes might be checked, but two of the three would be. And in certain cases, we have seen yield improvement as well. In terms of what is in it, I cannot really divulge the secret ingredients, but all I can tell you is that if you open your pantry right now, I'm pretty sure that you have those ingredients.
Okay.
So very common ingredients, very edible. I mean, if you eat it, it's not going to be big.
Yeah, I was just thinking, did you patent it? Because if somebody want to start a company, then what will be the process to get that patented thing to use? Is this something that you will supply, or is this something that they will get there and start, and then they will give some royalty or something? Whatever.
Right. I think, and this is a very important question as an entrepreneur that we need to really understand, is patentability. Since these ingredients were pretty well known already, it was very difficult to patent it, but we have a proprietary formula. The way we have approached this model are two phases. In the US, we have partnered with Reddy Ice, which makes ice and supplies. It is the largest ice making company in the country and in the world, apparently. And so they will directly manufacture the ice and sell the ice, functional ice. Whereas in India, we have a slightly different approach where we are making and mixing the chemicals ourselves, and then we will be supplying it to the ice manufacturers, whoever want to do it. And in fact, even before testing this ice manufacturer in Alibag, he's like, I want total, I just want functional ice for this entire region. And so he's just gonna get our chemical and just do it. So we are just supplying chemicals. So two ways to do it.
Okay, so basically you will supply the mixture of chemical, and then any ice maker can use that.
Yes.
To make the ice.
That is very easier model, and all you have to in the supply chain, you just need a smaller volume to just transport.
Okay.
Yes. For I think Banesh. No, yes. I think for Nagamurli, there is one question from YouTube, because you know that our program is also streamed live on the YouTube. More than some 70 people now they are there on the YouTube. Nagamurli, there is one question. Can you hear me?
Yes, sir. I'm here.
Any report of combination of fingerlings from Hapa method and sex reversal in RAS to get the best of both ends? Combination of fingerlings from Hapa method and sex reversal in RAS.
No, sir.
Can
actually,
comparison of that.
Yeah, actually, the deciding factor here is the eggs. Like when we are doing Hapa method, like when you are collecting, as I said, in natural, the eggs whatever laid by the fish, it takes into its mouth, it incubates there, and it releases the fry. So 100% of hatching is happening. All the unwanted things also will come. The non-growing, non-performing fry also will come. But when it comes to the egg incubation, like when we do that, 80% of the hatching will happen normally in the egg incubation. So the 20% which are lost, mostly they are weak ones, weak eggs. So we are eliminating the weak eggs there. Now, when it comes to the egg incubation in RAS, so the parents will be in a cement tanks where their fecundity will be very less, and those are the best eggs. That's why I said if the egg collection is done in the RAS, and if that sex reversal is done in the RAS, those fingerlings are giving 100% survival and the best performance, because here the genetic principle of the survival of the fittest is actually working. That is the only key there. There is nothing else. The survival for the fittest, whatever is getting selected in the RAS system, is the key success story of the RAS hatchery. But again, you collect the fry from the Hapa and bring it to the sex reversal in RAS. Maybe you get a better growth. Like Amit might be knowing if he has gone to the Abon hatchery where they do the sex reversal or whatever in the RAS, where they collect the fry from the pond. And there they are getting 1 gram size within 21 days from the fry stage. So maybe like you need to apply that technology here, the same RAS technology of fry to fingerlings, where they are using an automatic feeder, and it's a totally good technology. So maybe if we can use that Abon technology of sex reversal and fingerling rearing in a RAS system, he might be getting a very good result.
Yes. So I think that is very interesting how to develop the hatchery for tilapia fish. In India, the demand for fish, tilapia, sex reverse tilapia, that is monosex tilapia, is rising day by day because there is a common preference of many fish farmers to release ponds. There are many farm ponds in every state. In Maharashtra, more than some 18,000, definitely more than 18,000. And everybody want to go for culturing the fish. And comparative to other fishes, tilapia is having a wide acceptance, available at a competitive price, 100 to 150 rupees. And therefore, many people, they want to go for tilapia in farm ponds. So this particular hatchery, establishment of hatchery, one of our student, Mr. Nitin Nikam, he is also trying to have tilapia hatchery. Already he got permission from government of Maharashtra to start the hatchery, and I hope within 1 or 2 years, I think he will come up with the tilapia hatchery, and it will satisfy the need of farmers, local farmers. One thing for all the students, those who have joined for this particular program, I know that it is being time is little bit extended time, but we don't have any other options because of so many things. Okay, that it is extended period of a time we are going. But what two things we have observed from all the three our guests? All the three, they are now the towering personalities in the fisheries sector. Uh, One is their love for the profession. Yes, whatever thing, whether they are in fisheries sector, Bhaneshwar is in forensic sector now, and Amit is basically in poultry science, but also working in fisheries sector, but diversified in poultry science also. So basic thing is whatever profession which you choose, okay, we must be very honest with that. Second thing which I have observed, if you want to be a good entrepreneur, okay, you must have a commitment. Okay, I saw yesterday I have given time to all these three our alumni, and they were present dot at 2 o'clock. Sorry, 4 o'clock. Yesterday we had the meeting 4 o'clock. See, the commitment is very important for if you want to be a good entrepreneur, your commitment will make the difference. The people, those who are committed, they will progress. People, those who have love for their profession, okay, they will progress. Okay, and we have three these towering personalities as the Foundation Day lecture series. वैसे तो सबका सुनने के लिए बहुत अच्छा लग रहा था कि सुनते ही जाओ, but you know you have the limitation of a time, and from Gujarat, Sajid has also joined. Yes, and in the chat, I think I will just see whether, yeah. What is your view on suitable species of tilapia in India, in particular gift tilapia? One small question I will take, and for Naga Murli.
Yes, sir. Like Oreochromus niloticus gift strain, which is available with the World Fisheries Organization. Like, I think we call it Malaysia in Penang, that World Fish Center is there. Now they have currently 18th or 19th generation of tilapia, which they say it grows to 1 kilo size in 6 months. This is the latest generation. I think that should be the best one for India.
Okay. 1 kilo.
Okay, thank you.
In 6 months, they are saying the latest
yes, very nice.
1 kilo in 6 months.
Very nice. Yes, very nice. If you can get that one, the things will change like anything.
Yeah.
So I will invite a small comment at the last from Baneshwar Singh, then Naga Murli, and then Amit, and then we'll end up our meeting with the vote of thanks from our Honorable Associate Dean Dr. Shingare sir. Last comment from all the three dignitaries.
Thank you, sir. I think it was nice seeing you all, and thank you for the invitation. I think I enjoyed because it's after a long time I see, and I did not see you to have any change. I think I see some people changed, but you look like same 20 years ago. So that's kind of incredible.
Thank
I
you.
see same thing, and many friends and seniors, Bharat, Sandesh, and all my teachers still there, and
Yeah,
seeing.
Bharat, please unmute me also.
Yeah, so I think seeing all Santosh and everyone is really nice. I think it was good experience, and it's really refreshed all my memories. So thank you for invitation. And regarding the interpreter, I know that some of the things that that was not exactly in aquaculture side, but I feel like
Yes,
again
yes.
the techniques are similar, and will be happy to help anyone who need to use the techniques I talked about in my talk. Also, like regarding entrepreneurship, it's not only the starting a company. Like right now, I'm talking to a company called Illumina Technology. So they make a sequencing, right? So they have a kit, and we are collaborating in making their kit better. So like the body fluid ID, so we are trying to implement the method we develop in their kit, so that all over the world, whoever will use the DNA typing kit, they will have some part from my side. So the university also have like should encourage people to do, you know, to preserve the intellectual property. Like at my place, what is happening is every 6 months somebody come and ask, are you going to publish something? Don't publish, let me see if it has an industrial value. If it has an applied value, don't publish, keep it there, and then first we'll talk to company, and then if it's not good, then you publish. They It takes around $50,000 to patent one thing, and they are ready to pay because they know they will get more than that. So I think we have such a lot of innovators in our society, in college, and sometimes we just, you know, just don't think in that way, and we lose our, innovation comes at a small scale also, but sometimes we just put it there in like just publication, and then we are happy, but then we miss the trend, and somebody else pick that small part and make something good. So I think yeah, at the college level also there are possibilities of you know innovating and collaborating with companies to make things better. In terms of it's like in aquaculture, I think there are lots of research needed in metagenomics. I did not see lots of paper on metagenomic part. So any research that requires metagenomics, whole genome, like whole metagenome sequencing or 16S or 18S for any study. And if, like I have in my lab, next generation sequencing. So I can do anytime any sequencing is needed. And any times you have some sample and you not do okay, what may be the, microbiome, and there are thousands of bacteria, and I can be helpful in those situations. Also, I think there are people who want to go for higher studies. If they need some guidance or something, I will be happy to guide them. Like for example, it's not subject wise, and sometimes we have to listen. What happens is, like when I was applying, I had a friend, like a Indian professor from Andhra in US, and I talked to him, and he said, "You see, you have a great, background." And I was trying to get the molecular biology, but he was in different area. And then he said, why not you go to, aquaculture and molecular biology together? That will be very good for you. At that time, I thought, okay, he don't want to take me, so he's just giving some suggestion. But later, I felt like I would have done much better if I would have continued biotechnology and aquaculture together, right? So it's not like I did not do good, but that was a good suggestion. At that time, I did not take it positively. Right. So if anybody, and sometimes we don't have, guidance. And I started, in college, I started knowing GRE and thing, but there are possibility of getting trained and then go back and work for the development like Amit is doing, and not going, even if you stay, you can do lot to, and that's what the China did. They did lots of things by getting, training and then putting it back, right? So I think if for the students, anytime if they need, sometimes, they are a group of students or maybe final year, BFSC students, if they need to know how to communicate, how to, go for admission, if they want to go for higher admission and things like that, when to start, what should be done. I think I can be, available for them if they need help. So I think, or any other, I think, although I'm not working on this, but anytime if any, anything is needed from any alumni, I will be happy to help in any developmental work or anything that college want to do. So that's, and for the students who is working here, I see there one difference I felt when I came to US, and I want to share experience, is the work they are doing here, they want to make sure it works 10 times. Like I did my, in master and some other, research, what happens, we got the result and then we say, okay, publish it. What if next time it will not come like same thing? Okay, just do it and then let it go, publish it. So what happened when I came here, my PhD mentor asked me, okay, this worked, okay, add 10 more species, see what happens. I said, it's already done, no, add more. Somebody will question, you do more. So you do all validation before you go so that your method will work every time. Not like, okay, this is working, that's fine. But what if something will change? Will that work? If that will not work, then you will get stuck in the future. So it's better you do the things that somebody will, question you. You do it yourself before you go out for publication. Second thing is be confident on whatever you are doing, because that one thing I liked in doing my master. I was not confident on my own research, like I was doing, but I was not confident. If my result is not matching with someone big, I start doubting myself. Okay, maybe I'm doing something wrong. Maybe, I did not identify species correctly. And then I had a similar situation when I came to US. I had a small paper I submitted to a journal, and my result was totally opposite to a big group of 5 people from National History Museum, London, Denmark, National University of Singapore, North Carolina State University. They had multi-million-dollar, multi-nation collaborative grant, and my paper was against their paper. And when we submitted, my PhD mentor said, submit it to same journal and mention that paper and said that our result don't match. I was so scared. He said, you did it. You should be confident on what you did. So be confident, and then we are fine. We don't care what other people have. Sometimes it's not like a good or bad. They also did good, but sometimes mistake happen. But why to do second mistake by saying, okay, these are the big people. Don't say that, because then we'll get stuck. And the paper went to the same people who published. Out of 5, 3 said, good, it's okay, it happens. And 2 said, just collaborate. Can you collaborate with us to find out why our result is different than yours? And my PhD mentor said, no, that's their problem. We have to do, and sometimes you have to be bold with the editor. He said, you either accept or reject. We want to get decision within a day. We don't have a time. So what I was trying to say is, when you do research, be confident on what you are doing, and don't worry whatever, US, Europe, anywhere. If you are sure what you are doing, then you should be fine, and then only you can develop new thing, because new thing is not always same as something already developed. So be confident on what you are doing, because nobody knows better than what you do on your research. So that's some suggestion to the students.
And
thank you, everyone.
Any takeaway points? Yes,
and I'll be happy to help anyway.
From your
thank you all my teachers and friends and
I will
junior.
request Amit. Amit, are you there? Amit is in hurry, but still I will request him to have some just comment. Yes, Amit. Amit, please.
Well, thank you so much for inviting me for this wonderful, wonderful occasion. Congratulate the College of Fisheries for holding the mantle high, and for all the 39 years. I have enjoyed every bit of the every day that I was in college, and it's very nice to see every one of you guys, my teachers, my professors, seniors and juniors as well. I can tell you one thing, that Fisheries College and the friends have become a family, and it is one of the most wonderful experiences. I would be happy to be part of any research projects, or if you guys want any help from me, I would be more than happy to help you out. Most important thing I would tell our students is that gain knowledge and use that knowledge and adapt to these situations. You can become an entrepreneur by yourself. You can start a small business if you need to. You can do a job just like I am doing, and you can have your own company on the side and run it on Saturdays and Sundays. And that still is fine, but be an entrepreneur. That little bit of money that you can bring in home, it might not be a whole lot, but the pride that you have in making something unique and serving the community is tremendous. Thank you very much.
Thank you, Amit. Thank you very much. Nagamurli, your quick comment, please.
Sir, thank you very much for giving the opportunity to me, sir. And it was a pleasure for me to share my thoughts. And well, for my juniors or anybody who is wishing to enter into this aquaculture business, it might be tilapia or any other, I'm always ready to help them by all means. And personally, I'm looking to invest in India, especially in tilapia hatchery. So if anybody from Maharashtra is interested to do a JV with me, I'm very much ready to do that.
Yeah.
So I will be always indebted to College of Fisheries Ratnagiri, which gave me a golden path. Thank you, sir.
Thank you, Naga. And before I conclude this particular session, I must thank all the three persons, our three guest lecturers today, and organizing this Foundation Day lecture series is really a very charming moment for me. Those who have faith on this particular institution which makes you in a life, which provides you bread and butter, and when it comes a time to give returns to this particular organization, we'll have to remember all those important days, and its Foundation Day is one of such a day, and therefore we are very enthusiastic at our college level to organize this particular Foundation Day series. There are many opportunities. I think this particular Foundation Day lecture series has created, with Banish was saying, we will have some collaboration with the BSF. That particular project, Dr. Nitin Sawant, he will definitely take some things forward. With I think Amit will be interested in the functional eyes, and many collaborations, I think, with the functional eyes. And very nice to hear that Naga is going to come back to India. And your expertise is, I have already received 2-3 messages. Please provide me the number and email of Naga Murli ji. So definitely I'll be in contact with you. So the future is very, very bright in coming 5 years. Like we were discussing yesterday, Prime Minister Matsya Sampada Yojana is going to create lots of opportunities, and these particular opportunities will be harvested by our own students, our graduates, our postgraduates. And if they can do it, there will be many entrepreneurs, and we'll achieve all our targets. Let it be quantitative, or let it be in terms of value or quantity. No target is such that we will not achieve. There are many possibilities. And with this, I request our Associate Dean, Dr. Shingare sir, to kindly express the vote of thanks for the today's function.
Namaste. Good morning to Amit, Manishwar, and Nagamurli, and good evening to rest of all. It is my privilege to propose a vote of thanks and acknowledge the contribution of all dignitaries, the special invitees, participants who made this 39th Foundation Day a memorable. I, Dr. Prakash Shidgare, on behalf of College of Fisheries and Faculty of Fisheries, Ratnagiri, and the entire fraternity of the Institute, I extend my most sincere thanks to Dr. Chai Krishna Jena sir, Deputy Director General, Fisheries Science, ICR, New Delhi, who spared the time from his busiest schedule to grace the occasion and to guide the fishery alumni. This was the great opportunity to hear his thoughts, and this will surely going to encourage our fishery alumni, our staff in our future events. It is usually said that today's youth is tomorrow's nation. Our wealth is our students. And after the initial nurturing, the responsible young generation strive for the development of our society. The challenges faced during the studentship make them tougher to accept the challenges and reach to the new horizon. Our past alumni, Dr. Baneshwar Singh, Dr. Amit More, Dr. Naga Murli, they have set an example for all of us. To share their journey, they made it possible to join the celebration with a lot of affection and respect for the institute. Therefore, on behalf of the entire faculty, entire team, all staff of the College of Fisheries, Ratnagiri, I am very much thankful and grateful to Dr. Baneshwar Singh, Dr. Amit More, and Dr. Naga Murli for sharing their views on technical aspects with the fisheries alumni and our faculty staff members. During the last 38 years, the faculty has progressed and turned into a nucleus center for the development of fisheries, not only in Maharashtra State, but also national and international level. This was possible with the strong support and motivation of all former and present authorities. From the beginning of our faculty in 1981, the former Vice Chancellor, Dr. Savi sir, Dr. Kadrekar sir, Dr. A.G. Sawant sir, Dr. Mehta sir, Dr. Magar sir, Dr. Lavande sir, and Dr. Bhattacharya sir. All these former Vice Chancellor, they have given the special attention to this faculty of fisheries, as our faculty is very small faculty as compared to the other agriculture faculties. But then also they have given good attention and they have nurtured our faculty for the development. Also, I am thankful to our former associate deans. The first associate dean, Dr. Sankoli sir, who is then Dr. Rajesh sir, Dr. Kohei sir, Dr. Huku Joshi sir, Dr. Pai sir, and Dr. Hukum Singh Dakar. We have contributed a lot for the development of our faculty. I am very much grateful to our Honorable Vice Chancellor, Dr. Sanjay Sawant sir, for his dynamic support and active participation for the development of faculty. Since joining as a Vice Chancellor, he continuously in contact with the faculty, and he always says that we should not work with the only fishes, but there should be integrated farming approach. All faculties, that is agriculture, horticulture, and fisheries, should work in hands to hand. An integrated farming approach should be there so as to develop the Konkan farming community and the development of our university. He is always emphasizing on the development of the recent technology. In his speech and his guidance, in his address, he told that so many farm ponds are there in university campus. But out of that, only 2 ponds were utilized. And this year, he gave the directives to me and to our faculty that all maximum farm ponds should be brought under the aquaculture activities, seed rearing activities for the demonstration of the technology which is developed by the faculty of fisheries. So he is very keen, he is very interested in the fisheries activity for the development of these fisheries. So I am very much thankful to our Honorable Vice Chancellor for sparing the time from the busy schedule, because since morning 10 o'clock, he is continuously in meeting, and he just gave 15 to 20 minutes for his lunch, and he again joined our this faculty program. So I am very much thankful once again. I am also thankful to the all directors, that is Director of Instruction, Dr. Narkade sir, Director of Research, Dr. Aydankar sir, and Director of Education, Dr. Bhavesh sir, for their rock solid support and encouragement, without which it was difficult for us to achieve the excellence in our work, and also to organize this webinar at such a large scale. I am also thankful to Dr. Pradeep Hadonekar, who is the Associate Dean of College of Horticulture, Mode, which is in Sindhurga district, for his active participation and support for this webinar. I also thankful to our HODs, who have been fortunate enough to be backed by the team of very proactive and dedicated staff of our institute. Dr. Naik, Head of Department Aquaculture, Dr. Ashish Mohite, the Professor and Head Department of Fishery Technology, then Dr. Pause, Professor and Head Department of Fishery Hydrography, Dr. B.R. Chavan, Professor and Head Department of Fishery Engineering, Dr. Chaudhary, Professor and Head Department of Fishery Resource Economics and Statistics and Extension, and Dr. Swapnaja Mohite, Professor and Head Department of Fishery Biology, and their team, that is all associate professor, assistant professor, technical and non-technical staff members. I also thanks to Dr.
Hello. Hello. Sorry for disturbance.
Sorry, there was some net connection problem. So I am also thankful to the all extension department because they have taken very painful efforts to make this webinar grand success. So thanks to all of them. Then I am also thankful to our scientists from the research stations, specifically North Konkan region and South Konkan region. That is from the scientists.
5 घंटा 30 मिनट चला है.
Scientists from the Tarapurwala Marine Biological Research Station, Mumbai. Then from the Kharna Research Station, Panvel. Scientists from the Marine Biological Research Station, Ratnagiri, and Fisheries Research Station, Mulde, for their support, their continuous involvement and participation in this program to make it grand success. Not last but least, the students of our college belonging to different states of India. These students have contributed for the development of the fishery sector in government, institute, private sector, finance, self-employment. Dear students, the glory is because of your efforts on behalf of college, and I express thanks to all of our fisheries students, fishery alumni, the ex-students and the present students. The ex-students, specifically Dr. Deepak Chaudhary, then all other who are present in this webinar. I am very much thankful to them for participation. I am also thankful to Dr. Mangesh Shirdankar, the principal, Diploma in Fishery Engineering, who has also involved in this webinar and may participated to make this webinar grand success. I also thankful to Dr. Barve, which is our ex-faculty member and also the student of the first batch. He has also participated in today's webinar. I am also thankful to Dr. Barve for his involvement and participation in this seminar. So along with this, I am once again thankful to all of you who have participated, who have directly and indirectly involved and make this support, help us to bring this webinar to all of you and for make it a grand success. Thanks one and all. Thank you very much. Thanks.
Thank you. I think we'll end our meeting over here. And it was about more than 6 hours. So thank you very much. So it was more than about 6 hours of our discussions on the topic. So thank you very much for your participation. All the people, those who are joined for this particular program, and special thanks to all my 3 guest lecturers, Baneshwar Singh,